And hello, everyone. Good to see everybody. I think we're gonna get started as people join in for this conference, for this virtual conference. I'm gonna take the opportunity to welcome everybody on behalf of the American Society of Investigative Pathology. I am really honored to be here giving the welcoming remarks because this is one of the highlights of the society in support of the early career trainees. So for those of you who don't know me, I am Pilar Alcaide. I am a professor of microbiology and immunology at the University of Miami School of Medicine. And I have the privilege and the pleasure to serve as the ASIP president this year. And I am really, really, really excited about this conference. I want to start by highlighting the work of Dr. Sarah Dooley and Maria Antonia Zambrano in putting together this conference. And it's truly a joy to see how trainees evolve in this society and how you all get engaged with presenting your science and taking the opportunity to communicate with peers and navigate career opportunities and career challenges with each other. So I think the program that you're going to be listening to or attending to over the next few days is really tailored by Sarah and Maria and the committee and the rest of the people who supported from ASIP for you to really get the training experience of not only analyzing and presenting your data, but also to learn about all the disciplines that apply to pathobiology and you could perhaps incorporate in your project. I was very excited to see that a lot of abstracts were submitted and accepted, and that you are all going to have this venue to give oral presentations on your work and learn from each other. The program includes a lot of different aspects of pathobiology. I think it's going to kick off today with a very timely topic, which is AI. And I think that that's going to be that's like a great way to get it started and to see how we all need to start embracing this technology to really understand pathobiology and humans in health and disease. So I don't want to take time of the real important people of this meeting who are really you, all the trainees who are going to be presenting. And I'm really excited to attend as many sessions as I can. We all have busy schedules. And again, this meeting is meant to be for you to get all the learning experience. But I think you're going to have the opportunity to also present to faculty who will join on and off throughout these days. So with that, I'd like to welcome all of you because again, this is a highlight for an annual meeting. I would like to thank again the organizers, Sarah and Maria, and the SIP for their support in getting this meeting together. And I really look forward to learning from all of you and to contribute to your training in the best way that I can. So I think I'm gonna pass it on to the organizers of the meeting. Thank you and welcome.
Thank you so much, Pilar. So yeah, everyone, hi, welcome. We have a great meeting in store for you, I'm sure. PISA 2025 has been really fun to plan alongside my co-lead, Maria, and especially thanks to Anu, Anna, and Cole. We have been meeting since, I think, October now, really trying to navigate how to set up a virtual meeting, how to group all the amazing abstracts and everything in between. So it's been a real pleasure to work alongside every one of you. I also wanted to say a quick thank you to all of our speakers, our attendees, the moderators, especially to help us out throughout the next 3 days, and our special invited speakers. So we have a special invited speaker for each day. I think today we're going to be having a keynote about breast cancer. And then, of course, thank you to our ASIP staff. You can see all these beautiful virtual backgrounds were designed by Chelsea, I think submitted by maybe an Ingevic. That's what I would assume with the imaging. Um, and Lisa has been really our power of the background platform of Zoom, getting all of our ducks in a row and really organizing that. And Dr. Coleman is always there for us and guiding us with the best possible way to organize things in some capacity. So this meeting really wouldn't be happening without everyone involved. So, like I said, we have a lot of presentations to get through the next 3 days. So please pay special attention to our moderators. Lisa is going to have a little timer in the corner that you should be able to see. And if you miss out on an opportunity to ask a question while we have the allotted time, feel free to put it in the chat and the speaker should be able to respond in there. So thank you so much for your attention and logging on for the first day. I hope we get people to log on as much as they can and really enjoy a good meeting. So I'll pass it over to Maria, and thanks again.
Well, thank you, Sarah, and also thank you, Pilar, for the introductions. I just want to echo what Sarah said. Welcome everyone to PISA. It's been so much fun to learn, but also to organize this virtual meeting. Thank you everyone who submitted an abstract. It was an overwhelmingly amazing number of abstracts that we received. And thank you to the moderators as well. Thank you to the invited speakers in advance for the time that they're taking out of their obviously busy schedules for giving us and providing us with the knowledge that they bring. A special shout out to ASIP as well for giving us this platform of trainees to actually share our knowledge, share our passion in what we do for research. Shout out to the amazing Lisa, as always, the amazing Bill, and the amazing Chelsea for their support and everything technology, but also everything just like knowledge passed down, like generational wealth almost. So with that, I'm going to welcome everyone to the 3-day PISA virtual meeting. Thank you for taking time from your busy schedules. It's going to be a very, very cool amount of 11 sessions that we have with 50 speakers. So with that in mind, and given that we want to respect everyone's time, we'll actually start 5 minutes ahead of schedule. And with that, I want to introduce the moderators for session 1, which are Anna Tingler and we have Lu Huang, who are going to be introducing and moderating session 1, day 1 of PISA. So thank you everyone so much.
Hi, did I start? Okay, okay. My name is Liu Fang, and so glad to be here and to be the first moderator of the first session. Okay, let's welcome our first presentation, who will discuss the detection of ERBB2 copy number variants in breast cancer using whole exome sequencing. Please welcome Taylor Taser from Dartmouth Hitchcock Medical Center.
Okay, can everyone hear me?
Very clear.
And let me make sure my laser pointer's good. All right. So thank you for the introduction and the opportunity to talk about a project that I've been working on here at Dartmouth. So as I'm sure everyone is very well aware, breast cancer is one of the most common cancer types. The NCI gave an estimate for 2025 that over 300,000 people in the US alone be diagnosed with breast cancer, and there'd be an associated 42,000 deaths due to breast cancer. So even though this number is very staggering, the mortality has been declining as we've had better targeted therapies, one of them being for HER2, as HER2 positive breast cancers account for about 20% of breast cancers. So why do we care? So HER2, or you'll hear me throughout the presentation say ERBB2 and HER2. And so ERBB2 is the gene encoding the protein HER2. So this is a family of receptor tyrosine kinases, HER2 being one of the members. So this family is made up of proteins that are ligand dependent, where once they bind with a ligand, they'll be able to dimerize, phosphorylate, lead to activation of the RAS, PI3 kinase, and STAT pathways, leading to cell survival, proliferation, angiogenesis. HER2 is a little bit different though, as this one is the only one that's not ligand dependent. So it loses that regulatory aspect that the other HER molecules have. However, at baseline, HER2 has really low expression on the cell surface, so this isn't typically too much of a problem. However, in HER2 positive cases, we have amplification of ERBB2 leading to overexpression of HER2 on the cell surface. And HER2 is interesting in that it can dimerize with the other HER molecules, and then it can, say, for example, bind to HER1, and then you lose some of the ligand regulation as only one HER molecule needs that ligand. So it just leads to more aberrant pathway activation. Now with HER2 specifically, this is a target that has gained a lot of attention for targeted therapy. And there are quite a few FDA-approved drugs for the treatment of HER2 positive breast cancer. And there are treatments that can target the extracellular domain, the intracellular domain. It's kind of more up to the oncologist though, what kind of regimen. There are a lot of nuances to that have proven to be very effective at treating HER2 positive breast cancers, as well as some other cancer types. So this just means that it's really important that we can accurately and confidently detect ERBB2 alterations. And we have a few different methods that we can do that in the diagnostic lab. We have immunohistochemistry that can detect the HER2 protein. We have FISH that can detect the ERBB2 gene, and then what's been gaining a lot of attention recently is next generation sequencing, or NGS. So briefly, just to talk about the ESCO and the CAP guidelines. These are guidelines that have kind of standardized how we look at the FISH and IHC scoring to be able to call cases either positive or negative for HER2. But as you'll see, this is very nuanced. With our IHCs, for example, you'd see that it can be pretty subjective depending on the pathologist what qualifies as an IHC 2+ versus a 1+. So with a lot of these kind of more equivocal cases, they'll need to reflex to FISH testing in order to more confidently say, is this equivocal actually HER2 positive or HER2 negative? And with FISH, even though this is quantitative, this does also have a bit of bias and subjectivity to it. So with the FISH, you'll have a probe for HER2 or ERBB2. You'll have another probe for CEP17 or the centromere for chromosome 17. So in a healthy cell, you'll have two probes of each, or two signals of each in the cell. So with the guidelines, you'll look at the ratio of HER2 to CEP17, but you'll also look at the average HER2 signals per cell. So when we're looking at our amplified and our non-amplified, for our amplified, we'll say that the ratio needs to be greater than 2, and you'll need greater than 6 HER2 signals per cell. But with FISH, you can also have an equivocal if the ratio is going to be less than 2, and if you have between 4 and 6 ERBB2 signals per cell. So even though this is quantitative, it does still have that equivocal range. And as you can see here, sometimes it can be a little bit tricky to read the FISH. So even a quantitative method of detecting HER2 can still fall prey to subjectivity, and you really need an expert technologist to be able to read these FISH tests. And the guidelines for FISH, according to ASCO and CAP, are also very nuanced. You have to look at both the ratio as well as the ERBB2 copies per cell in order to make positive negative calls, but then you can also have equivocals that need additional workup. So all of this to say, it's very nuanced. To be able to call HER2 positivity, you need to do multiple tests, and both of the tests do have a bit of bias and subjectivity that can happen. So where does NGS come into play with all of this? NGS, or next generation sequencing, has a few pros that are pretty important that are very distinct from FISH and IHC. So with NGS, you're not just looking at the copy number alterations. You can look at other alterations that are happening in the genome. You can look at missense alterations. You can look, depending on the NGS platform, you can even look at methylation. So you can see if there are alterations that could affect the expression of HER2 besides copy number alterations. It's less subjective and biased than FISH and IHC. You lose that human aspect that can potentially affect your FISH and IHC results. It's more automated. It's starting to become more standardized. It's not in breast cancer yet, but there are guidelines for ERBB2 amplification with NGS in colon cancer. And then a lot of patients are already getting NGS performed on their cancers. So as long as you have the pipeline to look at copy number alterations, you might as well look at it at the same time. The cons, it's not very accessible. It's very expensive to get a sequencer. So you need to have that already built into the lab to be able to easily bring this on. And since it's decently new, it's slowly getting traction, but there isn't standardization and there aren't guidelines quite yet for ERBB2 amplification via NGS in breast cancer. The turnaround time is also longer, and then one of the things that can affect the quality of your calls is the tumor content in your sample. So these are just things to keep in mind. So we were curious to look at the quality assessment of some breast cancer cases that we had performed here at Dartmouth that underwent whole exome sequencing. And then when they were available, we wanted to cross-check our NGS results to our IHC and FISH results of these cases. So I want to emphasize when available because every case is different. Cases will get ordered differently depending on the oncologist or the pathologist who's working on this. So we took all NGS cases and then looked at if they had FISH or IHC. So there weren't any instances where we only had FISH or IHC. But some cases got one ordered, some cases got both ordered. It was a bit variable. And then just a reminder of the different call categories for IHC with positive, equivocal, negative. With FISH, we had it based off of the ratio as well as the copies per cell. And the NGS, we broke this down based off of the guidelines that are currently available for colon cancer for ERBB2 calls using NGS, where we had positive for copy numbers greater than or equal to 6, equivocal between 4 and 6, and negative as less than 4. So when we looked at the concordance between NGS and IHC, based off of the IHC score, we have our equivocal here at 4, and then we have positive here at 6. And we saw that especially at our negatives and our positives, we had really high concordance, only really rare discrepancy. So, but what's interesting is we did have quite a few equivocal results here that I'll get to in a moment. And then we saw very similar results again with our NGS versus our FISH results for positive versus negative based off of the ratio. We had really strong concordance with only a few instances of discordance or equivocal calls. So then when we actually break it down based off of the actual values, we again see really strong concordance between our NGS calls and our FISH calls. Only one instance of discordance here that was interesting, but we did have quite a few equivocal results that got called as negative with FISH, positive with FISH, and then with the copies per cell, we did have these equivocal ranges. So with these, we wanted to look at IHC to see if we could get a better idea of these equivocals, if they were actually leaning more towards HER2 positive or HER2 negative. And with these, what was interesting was when we had equivocals via NGS, we saw more instances where we had HER2 positives based off of our IHC results, making us think that having all three of these and having this equivocal zone that requires extra testing and just more in-depth look at these individual cases helps with being confident that we are fully catching our HER2 positive cases. And it's just going to show that an equivocal with the fish, we didn't have any instances where they were IHC HER2 positive, which we would have expected based off of the NGS also being negative, indicating that this equivocal with the NGS gave us strong support that we need to do additional testing. So with this, we saw that NGS, even though it's fairly new, it doesn't have guidelines yet for breast cancer ERBB2 amplification calls. We did have pretty good concordance with our FISH and our IHC in determining a patient's HER2 status. And using the guidelines that are currently available for colon cancer for our positive, equivocal, and negative gave us pretty strong, confident calls on the patient's HER2 status. And especially with the equivocal range, this really emphasized that we need to do additional testing to really make sure that we're confident if we're making a positive or negative HER2 call. And then, of course, with all our validations, really need to validate against IHC or FISH because these cases can be especially nuanced, especially with our equivocals. We can see that there might be multiple equivocals, one positive, one negative. So it really needs to have extra time taken care with making these calls, as this can affect what treatment regimen a patient will receive. And with that, I'd like to acknowledge everybody in the CGAT lab here, and then here are my references.
Thank you for the wonderful presentation, Taylor. And please direct any questions to the chat as our schedule is quite packaged. Yeah.
Okay, everyone, so let's move right on forward. And we're going to welcome our next speaker, who is going to be Janice Glover from Vanderbilt University Medical Center. And she's going to tell us a little bit about moving on from oncology to some infectious disease. So take it away, Janice.
Awesome. Okay, let me share my screen. Okay, can you guys see my presentation?
No.
No. Oh. Hmm. Okay, how about now?
Yeah, but I see your presenter view.
Ugh. Okay. Is that better?
There you go.
Ugh,
You can now see your screen.
what a day. Okay. Hi, everyone. So, of course, you've already saw my intro, but I want to thank the organizers for the opportunity to present. And I am excited to share some of the work I've done during my fellowship at Vanderbilt and kind of how I feel about the benefit of having automation in the clinical microlab. And I have no relevant financial or non-financial interest to disclose. And so in order to know how we got to automation in the lab, you have to know the foundation of the micro lab. And the micro lab is a critical component of healthcare and research. And I may be biased, but we're really dedicated to studying the different microorganisms, identifying infectious agents, understanding microbial behavior, and providing antibiotic results for treatment. So why choose automation? It really is broken up into two main things. We're seeing an increase in workload and then decrease in resources. So an increase in test volumes, complexity of patient population, need for cost-effective testing, and really a need for more rapid results. And then a decrease resources in there's a lot of retirements, decrease in personnel's entering the professions. The positions take a lot of time to fill, and then another lengthy process is just training. And so taking all those into consideration, what we hope automation will do for our lab and our patients is just a shorter time for colonies to appear, allowing quicker workup of isolates and just resulting in a quicker turnaround time. And so this will allow us to read the plates when they're ready to be read, and then just spend less time on really the growth of the cultures and allowing techs to focus on positive specimens, and then less variation in setup, right? So if we have a instrument that's consistently plating these in the same manner, we're just going to have less variation. And then the digital images really allows us to compare growth over time. So it's really important if we have like plate contaminants to see if there's anything visible at time 0. And that's been really helpful, especially for our workflow at Vanderbilt. Okay, so this brings me to WASP and WASP Lab. So here's a view of the current automation that we have in our lab. And on the left is WASP and WASP Lab. And so WASP stands for Walk-Away Specimen Processor. And then WASP is the fully automated specimen processing system where there's plate streaking, and then the WASP Lab is the incubation system. And that's where the pictures are taken over time for our microbiologists to review. And then we also have the Colibri on the right, and that's really designed for automated colony picking and then preparation of our microbial suspensions. And this is for downstream applications like antimicrobial susceptibility testing and MALDI. So the plating process in WASP performs really precise liquid dispensing onto the agar plates and ensures uniform streaking, like mentioned earlier, and this really creates colony morphology consistency among the different specimens. And this is really important for the next step, which is screening and reading. And so screening refers to the initial evaluation of the culture plates, so to detect the presence of microbial growth and just classify if we have single colonies, and really prioritize plates for further review. And so WASP Lab has these high resolution imaging that takes different pictures at different time points. And then I like to call this the reading area in the lab. I always go over there singing Reading Rainbow. So they always know I'm in the lab, and I love just kind of sitting back in this area and just seeing all the pretty bacteria that are growing. And then here's a closer look at the Colibri. And so this is an automated instrument that picks the colonies, and that's for the MALDI and AST McFarland preparation. And this instrument has been FDA cleared to use on our Vitech instrumentation, which is used for the MALDI and AST panels. And currently Vanderbilt's we're only doing our urine specimens on this right now. So just our MALDI, TOF, and AST helper, basically. So all the instruments and robots are fun and all, but the road to implementation and incorporating this into the workflow is a beast. So for WASP and WASP Lab validation process, we first took various specimen types and then plated and incubated them using manual methods as well to have them as comparison. And so we basically we're taking the WASP and WASP Lab plates, And since they're in those incubators, it's considered a smart incubator where the growth is really expedited. And so we had the WASP lab take images at various time points, and we really kind of wanted to see where's the best time point where we get really good growth. So yeah, this is kind of going into the validation process, which was pretty challenging. But initially we looked at various different samples, 110 specimens, and they had the picture read times at 10, 12, 14, 16, 18, 24, 36, and 72. And then each time point was evaluated on whether the culture was ready to be worked up. So meaning, do we see individual colony growth that can be used for identification in AST studies? And then in here, I don't have our urines, but we did incorporate that into our study. Just urines are a little bit more complicated with quantification. But nonetheless, all of our, it's kind of trending to where all of our specimens, a lot of them are ready to be worked up before 24 hours, which is the recommended time length. And so since we're in those smart incubators and the growth is occurring earlier, we kind of wanted to explore this a little bit more. So before we looked into earlier time points, the clinical lab is all about what are regulations, what are recommendations that we need to do since we're working with human samples. And so for MALDI, it states 18 to 24 hours of incubation time is really optimal for organism identification. And so none of the commercially available MALDI-TOF systems account for reduced times using smart incubators. So since there's limited data, we kind of wanted to explore this more, and that's kind of where I came in and really incorporated, my knowledge into this project. And so we took frozen clinical isolates that were previously placed on the MALDI for identification, and then placed those on the WASP and incubated them only at 10 and 16 hours. And at each time point, we performed MALDI and then also AST testing, and then also had parallel manual incubation just to compare results. And I won't have enough time to go over our AST data today, but each organism that we tested resulted in the acceptable ranges for essential and categorical agreements. So for the MALDI validation with WASP Lab, we wanted a wide range of organisms to test on at earlier time points, so a total of 222 isolates from different phyla, which encompasses some yeast and then a lot of gram negatives and gram positives. And I know it's really hard to see, but this graph just includes all the organisms that we examined. So gram negatives, we looked at a total of 83 clinical isolates, and they gave great identification at 10 and 16 hours, of course, except for Acinetobacter radioresistance. It gave an ID at 10 hours and not 16, if that makes any sense to anybody. And then Igonella only gave an ID at 16 hours. And then for our gram positives, they were pretty much perfect. They provided identification at 10 and 16 hours. The only thing, our arch nemesis is piney bacillus. It just kind of pops up everywhere, usually as a contaminant. Very hard to ID with our MALDI. So we did get an ID, but only at 16 hours for that one. And then we screened a small pool of yeast, and they all received 100% MALDI ID at 10 and 16 hours. And so based on that data we acquired, we chose the following read times for our cultures at Vanderbilt. So respiratory and joints are going to be read at 16, 24, and 36 hours. CSS are read at 16, 24, 48, and 72. And then our bloods and body fluids are read at 10, 16, 24, 36. And then urines at one time point at 16. In deep wounds, superficial wounds, abscesses, and tissues are read at 16, 24, and 36 hours. So then looking from like a quality standpoint, which is really important in a clinical setting, is to see, well, we've implemented the lab automation. Has it had an impact on our turnaround times? That's just the main thing. Like clinicians want results as fast as they can get them. So we went live in February of this year, and kind of compared to the months in 2024, there is a decrease in turnaround time shown by the median. And then looking at urines, there a similar trend is seen when we went live in 2025, February of this year. We see a decrease in our turnaround time, which is great. So of course, there are associated limitations with this study, and the first being just the focus on the WASP lab COPAN instrumentation. So the study uses WASP lab and it's a specific platform, but there are other applications. So other automation systems like BD Keystra that, has a very similar but different way of plating. So it'd be interesting to see like how they do with earlier time points. Of course, the frozen stock selectivity for AST, didn't show that, but I'm taking frozen stocks, that we specifically freeze because they're resistant. So the AST study was a little bit skewed, definitely prone to more of the resistant bugs. So it'd be more interesting to do real time studies, on more of those oddball bugs, just to see how they would do. And then, of course, the same thing for our identification using those frozen stocks as well. It's just a limited range. And it's always important to note that just the clinical impact, that's really why we're doing what we're doing and how automation can better help with the following. So just faster time to targeted therapy, improved patient outcomes. It just enhanced antimicrobial stewardship. It really has opened up the doors to kind of have a better communication with just getting these results out to our clinical team, and then lower healthcare costs due to quicker turnaround time as well, is always a good thing for everybody. And with that, I'd just like to thank, of course, ASIP, PISA. You guys have always been very instrumental in my whole journey. And so I really appreciate the opportunity to speak today. Of course, my attendings, program directors for the CPEP program, Kendall, David, Romney, and Lily, and then of course the leadership of the microbiology team. They've really been helpful with the time points, because 10 hours and 16 is it's not the best time when you're trying to get data. But anyway, and then of course funding sources, that I was able to receive a grant to fund this research. And then of course I'll be happy to take any questions. Thanks guys for bearing with me.
Great talk, Denise.
Yeah.
Same thing, everybody, just direct your questions to the chat.
Okay, thank you, James.
Thank
Our
you.
next talk, yeah, thank you. Our next talk highlights how deep learning applied to HE slides can stratify hepatocellular carcinoma, providing a non-invasive approach to tumor classification. Let's welcome Taylor Yosaka from the University of Pittsburgh.
Yeah, thanks so much for having me. Are you all able to see my slides?
Yes.
Okay, sounds great. Yeah, so this project is trying to use a machine learning approach, deep learning, to try to classify hepatocellular carcinoma, kind of for, ultimately for clinical purposes. So I'm just going to kind of give you an overview of the progress on this project to date. And try to bear with me through some of like the technical stuff. I will get to kind of like the application of this in the second half of the presentation. And I'll try to like just focus on the high level important things rather than getting too bogged down in all the technicalities. My screen, I can get my screen to change. Is it because I have my Zoom? Is that why it's like not responding to my clicks? Oh, just kidding. All right. So just for one slide, brief background. Liver cancer is the leading cause of cancer deaths around the world, and 90% of these are hepatocellular carcinoma, or HCC, and this will typically occur in the background of chronic liver disease. These patients have very poor prognosis and limited therapeutic options. So this is an area of need for better options. Yeah. So there are a few different ways that HCC is classified, and I should note that none of these are currently used in a clinical setting. But on the research side, one of these was developed by Eugene Hoshida. So these are the Hoshida subtypes, and broadly they're divided into 3 groups. So they are S1, S2, and S3. There are some different like histologic and molecular features that are associated with these, different pathways that are active. But at a high level, S1 and S2 are associated with worse survival, while S3 is associated with relatively better survival, and that will be something that we circle back to. So basically, to start this project, we wanted to see whether these subtypes have any kind of a spatial component. So they're based on bulk RNA sequencing, so it's basically a clustering kind of method. So they published in the original paper gene signatures associated with each subtype. So what we did was we took those gene signatures and we just looked at the spatial expression of those signatures in a Visium spatial transcriptomic data set. So that's what you're seeing here. You're seeing an example on one of those slides for each of those 3 signatures. You're also seeing like some cell type deconvolution that we did on those cells or on those spots. I won't go into too much detail about the results here, but basically there are spatial patterns. There are kind of interesting relationships between the different signatures, and also they have some kind of interesting relationships with the different cell types. I'll just mention that the S1 signature seems to be more associated with the non-parenchymal cell types, and S2 and S3 seem to be more associated with the hepatocytes. But yeah, so that kind of laid the foundation for this approach that we wanted to take. So we wanted to see if we can predict these spatial signatures from corresponding H&E, right, hematoxylin and eosin whole slide images. So this is a schematic of just the overall approach. To not go too much into the details, basically, the Visium data has these sequenced areas, they're spots, and then there's accompanying H&E. So we basically can tile the H&E into regions that correspond to each of those spots. And then the machine learning task is to try to predict the spatial gene expression in each spot from the corresponding area in the H&E. So each slide basically gives us a lot of different training samples that we can try to predict. We do some processing both on the spatial transcriptomics and on the H&E. And we train 3 different models. So there's 3 signatures, right? So we train one model for predicting each signature, and those models are trained independently. So I have one slide on the results. Basically, when we do an 80-20 training and test split, we get AUROCs ranging from 0.92 to 0.94. And you can just see at the bottom some examples of the similarity between the actual ground truth labels that we used and then the predictions generated by the model. So you can see that it is able to predict these in a reasonable way. So the more interesting application of this is to use it on to use it for some kind of clinical purpose, right? Just predicting arbitrary gene expression patterns is not that useful in and of itself. So what we do is we take this and we apply it to the Cancer Genome Atlas first. So basically in the training cohort, right, we have spatial transcriptomics and we have H&E. What we have in TCGA is not the same. So we have H&E, that part is the same. And then we also have some molecular and clinical information, but we don't have the H&E. And so we don't have the spatial transcriptomics. So we're not going to directly test those, like the ability to predict those labels anymore. What we're going to do is we're going to take the model, apply it to the H&E and generate predictions. And then we're going to see whether those predictions are useful for other types of analysis. So this is basically how we apply it. I apply the models to TCGA. So again, there's 3 models. So that's why you're seeing these 3 rows here. So for each model, we generate, kind of like a per tile prediction value, and you can see that here. And then what we actually do is we just average those across the entire slide. So we're just generating a single score for each patient for each of the 3 models. So basically each patient would have an S1 score, kind of like a predicted S1 score, predicted S2 score, and predicted S3 score. And I'll just point out that all of this is also based completely on the H&E. So there's no spatial transcriptomics or like bulk RNA that's going into this calculation. It's just the H&E. Once we do that, we did some principal component analysis just kind of for visualization here. So you can kind of see like the distribution of each of these scores. So each dot in these PCA plots, each dot is a patient, right? So you can see like basically the S1 scores, like they kind of increase in this direction. S2 goes in this direction, and S3 goes in the opposite direction to S1, actually. If you'll notice, those arrows are almost opposite to each other. And then, yeah, this is quantified here. So showing that like the Pearson correlation between S1 and S3 is negative 0.9, so they're very strongly anti-correlated, meaning they actually capture a kind of a lot of redundant information. So basically, we wanted to find kind of figure out how to group these in a way that's interesting, right? So just like the S1 and S3 models are very similar, so we don't just want to take the outputs from each model and like say the highest scores of each model, we like label it a certain way. So we basically did some clustering. I'm skimming over some of the details here, but we end up with these 3 clusters, A, B, and C, and we're calling this hepatocellular morphologic subtypes, or HMS. So these are based on these on the predictions generated by the model, but they're not equivalent to it, right? And so you could also say like these have some relation to the original Hoshida subtypes, but they are also distinct from the original subtypes. And just to drive that point home, you can see this PCA plot where you can see the Hoshida subtypes like projected into the same space. They look pretty randomly distributed, so they are quite different than these HMS subtypes. So the first thing we want to look at is whether this has prognostic ability. So we're looking at here overall survival, progression-free interval, and disease-free interval. These are different measures of patient outcomes, either actual survival or at least amount of time without recurrence of the disease or progression of the disease. And so we do see that these HMS subtypes are able to differentiate all these survival metrics, particularly with the HMS-A, those patients having the best survival outcomes. And that is the group that is closest to the like Hoshida S3 subtype, and those were the patients that had the best survival for Hoshida as well. So there's kind of like it kind of does make sense based on what we know. But just to compare this to the actual Hoshida subtypes, you can see these are the equivalent plots below here on the same patient data, but with the original Hoshida subtype annotations. And you can see that those are able to have some small ability to predict survival, but you can see like a lot of these lines are basically on top of each other. So showing how like these HMS subtypes actually are able to better separate patients in terms of outcomes. We also did some univariate and multivariate regression just to look at clinical correlates of these subtypes. And I won't go into too much depth, but like the HMS-A group, these patients have associated with like lower pathologic stage, better ECOG score, better Child-Pugh score. These are like different clinical measurements for either liver function or just overall functional ability. So it makes sense that those patients have better survival, actually. There's also hepatitis B etiology was associated with the HMS-A group as well. Then turning to molecular correlates, we looked at mutations that were associated with each one. And there are mutations, as you see here, that are associated with each group. I kind of have like these boxes to help you interpret the plot. But the main takeaway here is actually that we didn't see any of the big mutations that we look at in HCC, like the most common ones like TERT or P53 or beta-catenin, which is interesting. And to me, it just supports the overall hypothesis, which kind of, for me, was driving this overall project, which is that just looking at single mutations alone doesn't really tell the full story. So maybe looking at the H&E, the morphology via H&E is telling you more about the actual kind of clinical course of the patient. So this is looking at the bulk RNA sequencing that's available in TCGA. So basically, has some really interesting like themes for each subtype. So for the A subgroup, these had enrichment for a lot of metabolic pathways, really things that you find in healthy liver. So it's consistent with the idea that maybe these tumors are the ones that are more differentiated, retain more of their the healthy liver function. The HMSB are enriched with a lot of cell cycle related pathways, and then HMSC is enriched for a lot of immune related pathways. And actually, that last point was maybe you want to look at a signature for immunotherapy response, specifically anti-PD-1 response in HCC. So we take this signature and find that it's actually strongly enriched in that HMSC group, which is that kind of immune annotated group. So kind of an interesting hypothesis that maybe these patients could be best responders or best candidates to receive immunotherapy, and this is something that we're following up in more depth right now. The last thing I'll show you is just validation that we performed. So, just to make sure that these results are generalizable outside of that TCGA cohort. So this is a cohort of 50 patients here at the University of Pittsburgh. And you can see on the left, we have a nice distribution of patients across the 3 groups. And I'm just showing on the right the equivalent plot for the TCGA patients. But then we looked at the overall survival in these patients, and we found that indeed we see the same trend where HMSA has the best survival outcomes compared to HMSB and HMSC. So just to summarize this project, we basically have developed a method where we can take these H&E whole slide images and use some machine learning and kind of some other reproducible methodology, and we're able to stratify these patients into 3 groups, which have unique molecular phenotypes, unique clinical characteristics, and also importantly, unique survival outcomes. And so hopefully we can continue to work on this and develop it into something that can be used in the clinic. Thanks. Yeah, and thanks again to ASIP, all the organizers, also to all the funding, and also my graduate program here for all their support.
Nice talk, nice talk. If you have any questions, please direct to the chat, and let's welcome to the next speakers.
Great job, Tyler, once again. Let's shift to our next speaker, and that's going to be Dana Julian from the University of Pittsburgh, and they're going to be talking to us about their work on Alzheimer's disease.
Dana
Oops,
might
didn't
be
realize
muted.
I was still muted. Okay, does everything look okay? Okay, good. Okay, hi everyone, I'm Dana. I'm a graduate student with Dr. Julia Koffler at the University of Pittsburgh. And we're looking at white matter hyperintensities in postmortem human brain samples in the context of Alzheimer's disease. So, white matter hyperintensities are radiologically defined regions of often thought to be of myelin loss. They lead an individual to be 2 times as likely to get Alzheimer's disease, 3 times as likely to get a stroke, and correlate with Alzheimer's disease onset, progression, and severity. However, we know very little about what these lesions look like at the tissue level because they're hard to find in postmortem human brain tissue. And so there's a gap in knowledge understanding histopathologic signatures of these clinically important regions. We've overcome this limitation with an ex vivo postmortem MRI pipeline. And so now my project is taking a deep dive into the histologic characteristics of these lesions with digital pathology and machine learning. So the overarching goal is to identify tissue features of white matter hyperintensities that correlate with increased rates of cognitive decline in AD. And we have clinical information on these individuals as they were followed by the Alzheimer's Disease Research Center at Pitt. Our overarching questions are: how are cellular neighborhoods and tissue architecture altered therein, and how might this contribute to clinical features of disease? Specifically for this presentation, I'll be focusing on oligodendrocytes and myelin. So these are white matter pathologies, which are characterized by high oligodendrocyte and myelin content. It's so fatty that the brain tissue appears white. And this fatty tissue coats neurons, which help insulate neuronal axons so cells can communicate from one cell to the next. And I'll be talking about 4 main data types. First, Luxel fast blue H&Es, where Luxel fast blue stains myelin, this nice blue-purple color. Then fresh frozen samples from the white matter of human brain tissue, H&E tissue, and PLP1 for oligodendrocytes. And here's a graphical outline. I'll be talking about weakly supervised deep learning approaches, lipidomics on white matter, and then trying to bring these all together into a multimodal machine learning model to predict white matter hyperintensity, volume, and cognitive decline. So here's another schematic looking at our postmortem pipeline. So after autopsy, the left hemisphere of the brain is embedded in a sucrose agar solution, which is undergoes 7 Tesla MRI, and this is performed in a cutting guide, so we know at each longitude and latitude where we cut if there was a white matter hyperintensity at that locus. We don't use this to bias our sampling, but to inform our analyses. So we collect the same 6 standard regions of the brain from every patient, and these are regions of the brain that are most likely to have white matter hyperintensities. Although I want to point out there's a lot of heterogeneity in their volume, so they're marked here in red. You can see some are a pinprick size, where others are all encompassing of the sample and even extending beyond the sample where we collect. And here I gave a semi-quantitative grade to how many white matter hyperintensities were found in each location in our cohort and their severity. We have 85 brains that we've analyzed in the first cohort, and we're adding an additional 40 for a total of 115. So the first aim is to use weakly supervised deep learning to uncover regions of high importance in predicting white matter hyperintensity presence. And for this, we're using Luxel fast blue H&E images. I've been using CLAM, which is a deep learning model that learns to predict here slide labels. So it's a classification task. It breaks your image up into a series of tiles, clusters those tiles by how they appear and where they're located on the slide to reduce noise. And then after making its prediction, it uses an attention mechanism to decide how important that specific tile was in making the prediction. And these are left skewed. You can see this histogram shows the attention paid for all tiles in an image. And this is because this model was first built to recognize cancer. So of course you want to minimize benign areas on the slide and then amplify the signal coming from what is thought to be cancer. And that's very good for our purposes as well, because white matter hyperintensities often don't cover the whole slide. So here I used a feature extractor, which takes the image and turns it into a series of vectors that can be manipulated in the convolutional neural network. And I used one that was specific to brain images. So it was trained on 10 terabytes of data from whole slide images from all kinds of conditions, neurodegenerative, cancer, etc. And I trained 2 models, one that was trained on all tiles from the whole tissue, and then one that was trained on only white matter tiles. Both models did fairly well. The whole tissue model, its top folds, I trained this 5 times across 5 folds. All 5 folds did better cumulatively, or the top 5 folds performed better, but the white matter model did better overall. And here you can see the confidence of the model. So each instance, how confident it is on the x-axis. And so the whole tissue model was more confident in predicting the white matter hyperintensity cases, whereas the white matter only model was equally as confident in predicting the 2 cases. Here are some representative images of the heat maps that it produced. So it was really interesting to see that in the normally appearing white matter tissue, it paid a lot of attention to the gray matter, which might suggest some upstream neuronal dysfunction that might be leading to white matter problems deeper in the brain. And then in the white matter hyperintensity model, whether or not it predicted the slide correctly, it tended to pay attention to the deep white matter here, which is where white matter hyperintensities are generally found. And then when we force the model to only look at the white matter, we see kind of mirroring results. So it pays attention to the deep white matter generally in the white matter hyperintensity tissue, and then more attention is paid farther away from the lesion in the subcortical white matter in the normally appearing white matter samples. I extracted the top 10% attention tiles in the white matter only classification model and found that indeed in the white matter hyperintensity model, there was less myelin positive pixels as shown by LFP staining, which is what one would expect from previous literature. And so we were happy to see that in our work as well. I took a step back and I counted all positive LFP pixels across all 500 images, and I found that indeed there was less Luxel fast blue signal in the deep white matter, which is also what we would expect. And it was also interesting to see that the confidence that the model had and the attention it paid per region correlated with each ROI. So for instance, in the whole tissue model, when the attention was higher in the deep white matter, there was more confidence being paid to its designation. And now the next step is to look at these within the context of the radiology. So we have an alignment tool that will align MRI with coronal gross slab images, and then with 2-point registration, overlay our whole slide image, and then we can take the ROI from the MRI information and then overlay that on our host slide image, which we've been working within the QPath software. And so now we want to look at the heat maps and attention produced by the model within the context of the radiology. And so next, we're looking at the contralateral hemisphere to look at lipid alterations. So we took samples from the right hemisphere, and because white matter hyperintensities are symmetrical, although we don't have the radiology information from this hemisphere per se, we can refer to the other hemisphere for white matter hyperintensity designation. And these samples all came from the frontal parietal periventricular white matter, and this is the region which has the most severe lesions. So when we look at our volcano plot of differences between lipid species in normally appearing and white matter hyperintensity tissue, we don't see that many differences. Just a few lipid species were differentially regulated, but we weren't discouraged by these findings because we expected the lipid species to swell and shrink in large swaths more than have partially differentially regulated species. But those that were differentially regulated were myelin enriched, which was good to see as myelin covers 80% of the white matter. And then next we ran a PCA and found that it was could be easily to distinguish normal and mild samples from moderate and severe white matter hyperintensity samples, which I think goes along of the lines of the heterogeneity that we were seeing. So we expect that there would be lipid dysregulation in the white matter hyperintensities, but because some are so small, those that were mild, it's not surprising that they clustered with those that were normal. Then we looked at lipid saturation differences, and we found that across most lipid species, there was a huge decline in the saturation levels in the moderate and severe samples. And so this might point to differences in myelin stability, as lipid saturation in the myelin is really important for its structure and function. And then lastly, we made a random forest classifier and fed in information about the lipid species and found that lipid species alone were highly predictive of white matter hyperintensity designation. And then using Gini importance, we found that a lot of the lipids that were important in making that distinction, first were myelin-enriched lipids, and second, many were oxidized lipids, which would agree with one of the prevailing theories in the field that white matter hyperintensities stem from chronic hyperperfusion of the deep white matter. And then lastly, touching upon counting different cells and their cellular neighborhood architecture, we looked at cells by H&E and also by PLP1 in situ hybridization. So first, we started with PLP1 in situ hybridization because most, or really all, oligodendrocyte markers are not very reliable in human postmortem brain tissue, but we found this one to really highlight the nuclei oligodendrocytes really well. However, unfortunately, we're doing this all on an automated stainer, and we found that the signal was not reliable across the slide. So although this can be a great marker, in some instances, it wasn't reliable for our uses. So now we're training a model to recognize the oligodendrocytes in H&E images. So the plan is to do olig-2 staining for oligodendrocytes, scan in those slides where olig-2 works well, and then de-stain them so the H&E background is left, and then try and train a model using the ground truth of the olig-2 to recognize oligodendrocytes in H&E. And then we're bringing this all together in a multimodal machine learning model. So this is an example of one that was built from the Mahmood lab at Harvard. They brought together whole slide image data, cell graph data, which treats each cell nuclei as a node on a graph, and then creates a convolutional neural network of the neighborhood architecture to predict the label. And then the third mode, well, here it was genomics, which also brought in copy number variation. And then each of those modes, as you can see in this cube in the top right, each unimodal was treated as like a vertices on a Rubik's Cube, and then the bimodal product was the face of the cube, and then the trimodal product was the interior of the cube. And then this cube was used to predict survival across 14 different kinds of cancer, and it did better than any one of those modes alone. And so we're adapting that, but instead we're going to be feeding in LFB H&Es in one model, H&Es in another, H&E based cell graph data in one, and then oligodendrocyte cell graft data in another. And then instead of genomics, we're going to use lipidomics. And instead of copy number variation, use species quantity, saturation status, and carbon chain length. And then we're going to see how well this trimodal model does at predicting white matter hyperintensity volume and cognitive decline. And then one other really cool feature about this model is that it can give attention scores not only across the slide, but to each and every cell in your cell graph. And so we're interested to see if higher attention is paid to the oligodendrocytes like we hypothesize. And so just to conclude, LFB H&E images can be highly predictive in white matter hyperintensities, as is lipidomic signatures, and we look forward to bringing these all together into a multimodal model. And I'd like to thank my wonderful lab and my mentor, Julia, and all of our collaborators. I'm happy to take questions if there's time, but I have a feeling we're short on time.
Great talk, Dana. We're happy to direct any questions to the chat.
Okay, thanks.
Thank you, Dana. Very interesting talk. And our next presentation comes from Brigham and Women's Hospital, showcasing human iPSC-derived skin organoids that mimic cutaneous inflammation and injury, a promising step towards modeling complex skin pathologies in vitro. Please welcome Anthony Sheets.
Okay, thank you. I just need to get my talk set up here. Give me one sec. Okay, can everybody see my full screen? All right, let me just my pointer here. That's fine. Okay. So thank you everybody for having me today. These have been some wonderful talks so far, and really some tough acts to follow. So I'm going to share with you some of the work that I've been doing for the past few years as a postdoc in the laboratory for cutaneous stem cell biology and epigenetics at Brigham and Women's Hospital, focusing on growing skin organoids from induced pluripotent stem cells. So skin diseases are highly prevalent, and they affect about 25% of the United States population and about 1.8 billion people worldwide. And these account for significant morbidity, pain, psychosocial distress, and mortality in the case of skin cancers like melanoma and cutaneous T-cell lymphoma. But models that fully capture the features of native human skin are very challenging to achieve. And this year marks the 50th anniversary of Reinwald and Green's foundational work establishing keratinocytes as not only able to be cultured in vitro, but to also create stratified squamous epithelium in a dish. And really, this has served as a workhorse assay for the past 5 decades to study cutaneous biology. But one of the real challenges is the incompleteness of the models that we use for skin disease, both the two-dimensional ones, as I mentioned, they're three-dimensional cousins grown on scaffolds and with other cell types integrated into them. And there are important differences between the animal models that we use to model skin disease and bona fide human skin, as you can appreciate on the right side of your screen. For example, when we look at this biopsy of human skin in the bottom corner here, we can appreciate the epidermal reedy ridges, we can appreciate the undulating dermal papillae, where the capillaries arise to nourish the overlying epidermis. And we contrast that with the mouse skin immediately adjacent to it. You can see that the epidermis is much thinner. There's the lack of reedy ridges. And importantly, rodents include this muscle, the pedunculus carnosus, that really drives differences in wound healing between our species and theirs. Now, I'm not the first person to point this out, of course, but for these reasons, among others, including the low concordance between outcomes observed in rodents and humans, these preclinical in vitro and animal models have really been called into question, culminating in these more recent announcements, including this thing that's now called the Standardized Organoid Modeling Center that are going to be sponsored by the National Institutes of Health as we aim to move further and further from animal models and into more what are being called human relevant models. So organoids are self-organizing three-dimensional structures that are intended to mimic the architecture and physiology of the organs and systems that they're intended to recreate. And human skin organoids, as you can see in this schematic here, can be differentiated from pluripotent stem cells, and they really represent an important advance in the study of skin development and disease, because this is truly the first in vitro system whereby hair and other dermal appendages arise spontaneously. And you can see in this image in your upper right corner, this is what these look like under the microscope, under a dissecting scope. So when we modulate TGF-beta, BMP4, and FGF2 signaling pathways, as depicted in this cartoon, we end up with the co-induction of the surface neuroectoderm and the surrounding mesenchyme. And ultimately, then these develop as cystic structures that are surrounded by hairs. So this is basically inside out skin, and in many ways resembles a teratoma. If you've watched too much TV like we in our lab have, you think that these things really resemble a tribble held by Captain Kirk here. And I'd be remiss as a pathologist to not share with you then H&E images of these. So in some of these less mature skin organoids, we can see indeed that there is a squamous epithelial line cyst at the center here. It's surrounded by this mesenchyme that not only has developing hairs, but we've observed microvessels in here. There are developing glands, there's neural tissue that develops, and there's cartilage that develops in this system as well. We can confirm that these are microvessels that are growing here through immunohistochemistry for things like VE cadherin and smooth muscle actin to show the surrounding pericytes. And you can see that these are adjacent to a developing gland here. In their more mature form, then, they retain this cystic architecture, and you can see that the hairs have really matured here to look not unlike what we see in bona fide human skin. So these have a stratified epidermis, hair follicles, glands, microvessels. They're innervated. They have melanocytes that contribute to pigment formation. And as I mentioned, they form some cartilage and some muscle as well. But so now we're starting to ask the questions of how do we actually leverage this technology to study skin inflammation, such as dermatitic insult, as you can see in the upper right-hand corner here, and then injuries like decubitus ulcers. So most of the characterization that's taken place thus far has really been on what do these organoids contain, and now we're trying to move into the direction of what is it that they can actually do and teach us. So we have the following questions that we're aiming to answer. First of all, when do the endothelial cells and then the resultant blood vessels actually arise in the skin organoids? How does the vascular network evolve over time? And what are some of the molecular mechanisms driving vasculogenesis and subsequent angiogenic remodeling in this system? And then importantly, what's the actual physiologic capacity of the microvasculature? Can we actually add cytokines to show that we can activate the endothelium? And can we actually elicit reparative responses after mechanical trauma? So we've been leveraging a VE cadherin or CDH5 GFP induced pluripotent stem cell system where we can actually monitor the dynamics of vasculogenesis over time with fluorescence microscopy. And as you can see here, we identified GFP positive endothelial cells arising within the skin organoid mesenchyme as early as 6 days following the initiation of that differentiation process. We can use this technique to monitor how the vasculature evolves and grows over time. You could see in some of the lower panels that by day 40, we have these sort of aggregates of endothelial networks that are beginning to form. And at 77 days, they've now begun to interdigitate between those developing hair follicles. The vasculature really persists throughout the entire duration of the culture here. So we get to about 4.5 months and the vessels are still present. And we see these vessels forming in about 85% of our organoids. We wanted to understand what some of the molecular effectors were driving this phenomenon. So we used a multiplexed antibody array to determine soluble mediators of vascular and angiogenesis that are produced by the organoids themselves. And as you can see here, we've identified things like angiogenin, VEGF, and placental growth factor as being significantly secreted by the organoids themselves and increasing over time in culture. When we profile these immunohistochemically then, we can appreciate some stark differences between early and late organoids. So the vasculature in these early organoids starts out as being fairly restricted to some of these small islands in the mesenchyme surrounding the organoid. But in the later stages, where there are more hairs present, the vasculature has really developed to encircle the entire organoid and has a robust pericyte coverage. And we can appreciate that even greater at higher magnification, where we can see in the early organoids, there's really a paucity of microvascular pericytes surrounding these vessels and a limited amount of collagen 4 positive basement membrane, and that these features all increase with time. Importantly, these arrowheads are calling out vascular lumina that we've identified in here. And we've also seen a lot of material floating within these lumina that appears to be actually apoptotic cells. I'm not going to show you the data today, but all this is to say that we are able to form patent lumen containing vessels within these organoids that persist throughout the duration of the culture. So the next questions we wanted to try to answer, as I mentioned before, are evaluating the physiologic capacity of the microvascular networks within these organoids. So when we add pro-inflammatory cytokines like TNF-alpha, interleukin-1 beta, and interferon gamma, and evaluate these by immunohistochemistry, we can see that following 24 hours of treatment with these cytokines, we're able to actually activate the endothelium and the surrounding perivascular stroma, as evidenced by robust inductions in ICAM1 and VCAM1. And moreover, then, when we use a multiplex antibody array that measures 105 different cytokines that are secreted into the organoid conditioned media, we can identify VCAM1 as being strongly secreted by the organoids into their surrounding culture, along with thrombospondin 1. And in addition, when we profile more robustly the individual cytokine-dependent effects, we can see that there are unique profiles that arise on a cytokine-dependent level. For instance, when we add TNF-alpha, we see that that's the only cytokine in this system that induces CCL5 or RONTAS expression. When we add interleukin-1 beta, that was the only one that drove IL-6 expression. And when we added interferon gamma, we found that that was the only cytokine that induced IL-18 binding protein along with the B-cell activator activating factor. And many of the things that we've observed through this technique then are in line with other observations that people have made, not just in individual cell culture models, but in animal models as well. The last point I want to describe with you today is then the mechanical trauma piece. And so wound healing has been an interest of mine for a very long time, starting as a graduate student, and continues to be something I study as a postdoctoral fellow and dermatopathologist. So of course we wanted to try to answer the question of whether we could injure these organoids and stimulate healing responses. And indeed, that is the case. So when we take these organoids and sharply dissect them, either with scalpels or scissors in culture, we seed culture vessels with them where they're embedded in a matrix composed of type 1 collagen and growth factor reduced matrigel, which we can then supplement with growth factors such as VEGF. And again, using that same VE cadherin GFP reporter, we can monitor the healing responses over time. And as you can appreciate from these fluorescence images, then when we add VEGF to that culture matrix, we can induce a significantly higher number of endothelial sprouts that also grow out into the surrounding matrix to a much greater distance. And in addition then, one of the real powers of this system is compared to other in vitro platforms, we have nearly all the pieces of bona fide human skin present in the dish. And as you're seeing in these bottom panels here, we're actually looking at reepithelialization, the migration of keratinocytes following that injury. And so when we look at these at higher power, we could see the migrating epithelial sheets, and indeed we could measure the distances that those traverse as well. We have not yet put pro-epithelializing growth factors into the system yet, but that is one of our plans for future investigations. So to summarize, we've seen that vascular endothelial cells arise from the skin organoid mesenchyme in the earliest days of the differentiation protocol. These microvascular networks evolve from the mesenchymal condensations that surround the organoid to form vast arborizing networks that persist throughout the duration of organoid culture. The organoids themselves secrete multiple vascular and angiogenic growth factors and soluble effectors that we hypothesize are critical drivers of endothelial growth, tubulogenesis, and maturation. We've demonstrated that the microvasculature can be activated by pro-inflammatory cytokine treatments, and we're now the first group to show that these organoids are able to mount reparative responses to mechanical injury. So overall, our work establishes the skin organoid model as being highly relevant to future studies of dermal vasculogenesis and to study the roles of cutaneous microvasculature in immune activation and wound repair. So thank you for accepting my abstract to present today. I'm really excited to participate in the conference. I'd like to thank the labs that I work in and my postdoctoral mentors, along with my financial support, and I'm happy to answer questions through the chat and through email.
We have a question from the chat, like, have you stained for any mast cell markers in your skin organoids?
We have not, and as pathologists, so we use the H&E to guide us with everything that we do, as I was showing you. And you know, one of the real limitations of this system is that there is a lack of any immune cells that develop in here. Now I'll tell you, the fact that these have cartilage, and we've observed actually that the cartilage can ossify and form bone in here, does make us wonder whether if we carry out the culture long enough, we will see any sort of a marrow space forming. We have seen a few things that on H&E look like they could be extramedullary hematopoiesis, meaning blood development that's growing outside of a marrow space, but we have not identified any bona fide immune cells in there, either by H&E or by using CD45 immunohistochemistry to look for the total immune cell population.
I have another question from my own is how functionally mature are the microvascular networks, and is there flow or perfusion support? Like, yeah.
That's an excellent question. So the microvasculature matures over time. It gets greater and greater degree of pericyte coverage. It does appear that the number of luminal spaces increase. And the thickness of the basement membrane and degree of collagen 4 positivity increases over time as well. The proliferative index, which I didn't show you today, actually decreases as the organoids mature, which is in line with everything we know about pericytes as far as how they stabilize the microvasculature. These are not actually perfused, though. And that's actually one of the future directions that we have. So we're actually starting to collaborate now with Guillermo Garcia Cardena's group at the Brigham, just across the street from our lab, to start perfusing these organoids because, of course, the mechanical forces are so important for then that higher order specification of the vasculature. And so far, none of the studies that have identified the vessels in these organoids have shown that there's anything beyond, capillary level endothelial specification.
And we have another question from the chat, like how far are we from developing organoids with a full functioning immune system?
So I know that there was work published about a year ago where these organoids were co-cultured with macrophage-like cells that were differentiated from the exact same stem cell line to, prevent that immune cell mismatch. And in that work, the macrophage-like cells home to the abluminal surface of the vessels and served what the authors called a stabilizing kind of influence, and they showed that it altered the microvascular organization and so forth. I do know that there are other labs that have done derivations of immune cell differentiation from stem cells. I know George Daly's lab, for instance, is doing that kind of work. And kind of one of my dreams is basically to kind of put together a marrow organoid, if you will, in series with a skin organoid, grown from the exact same stem cell populations to see if we can fully reconstitute the immune system, because that would be such a huge advance for this model. But I think that would be really challenging to do.
And what kind of predictions would organoid modeling provide for organ-to-organ communication? And do you know if it is possible to generate multi-organ organoids for this purpose?
So again, like I was saying, I know of some people who are trying to do, a few different organoids in parallel and in series. And, in addition actually to the like a marrow organoid with skin, another important avenue would be to study things like, the link between like the gut and skin or cardiovascular system and skin. And we have a lot of evidence, for instance, that, in psoriasis, that actually can increase cardiovascular disease risk. And so the idea of studying, basically a psoriatic skin organoid and what it secretes and how that might influence a cardiac organoid, I think would be also a major advance for the field. So some of these things are in progress in my head and also, in the collaborations that we've established with the Garcia Cardena lab, but I think we're a few years off from that still.
If we don't have any questions from the guest, I will have another question.
Sure.
Okay. And I wonder, could this system be adapted for high throughput drug screening or regenerative studies? And what readouts like the angiogenesis indices or ECM remodeling are most robust for screening?
That's an excellent question. I think that's really the goal of this. Some of the original work, in fact, the original paper documenting the skin organoid system, successfully xenografted these to the backs of nude mice. So of course there's interest in, using that kind of hybrid system to study, skin grafting and wound repair in that setting. But I think really, as you're getting at, the power of having this system where you can generate something like 200 organoids that get up to about 5 millimeters in greatest dimension is to actually have them in, the kind of a screening model. Nobody has done it yet. I think a lot of people dream of doing it. As far as the readouts, I think one of the real challenges that we encountered when we were trying to develop our wounding model is what's really the right mechanism of injury for this, particularly because things are inside out. And when we think about wounds occurring a lot of times, we often think of them as coming from outside in, though that's not always the case. We know, ulceration can occur from things like vasculitis. We know that there are penetrating traumas where it can, kind of be, outside in and then inside out. We had tried to do a couple of, barotrauma based injuries with needle injection, but our needles were way too big. I think it's a ways off before we're at kind of a high throughput where, you can have kind of, some of these mechanized systems that have been described earlier today that are creating these injuries. And then, I do think though, like fluorescent reporter assays are really going to be, some of the strengths of the system.
Excellent, excellent. And thank you for the presentation, and thank you for Fabiana, and thank you for being there on time after class. Yes. Okay.
Thanks.
Thanks.
All right, moving to our last speaker, we have Fabiana from the University of Florida, and let's just kick her presentation right off.
Hi, thank you so much for the welcome. I'm gonna go ahead and share my slides. Just checking, can everyone see my screen? Okay. My presentation is about a project about bovine placental-derived bioink for reprogramming the tissue microenvironment for the enhanced regeneration and senescence reversal. And I wanted to give a shout out to my ASIP mentor, Menglu Yang, who I worked with during the summer at the Department of Ophthalmology at Harvard Medical School, as well as my project mentor, Dr. Mei He, in the UF Department of Pharmacy. So, to give a brief background about the rationale for this project, trauma wound care seeks natural materials that are biocompatible and can improve the regeneration of functional tissues and muscles while also reversing the damage-induced state that contributes to impaired healing. And the two components of the bioink that I was exploring through this project is collagen type 1 from bovine placenta, because we hypothesize that it can better replicate the human tissue microenvironment, tapping into placenta's known rich regenerative and biochemical properties, as well as extracellular vesicles derived from amniotic fluid, because past research has shown that they can be very anti-inflammatory, promote angiogenesis, and have a lot of regenerative properties as well as anti-senescent properties. So both of these components we use for droplet encapsulation. But before diving in deeper into that, I want to give a brief background about what extracellular vesicles are, in case some of you are not familiar. They're lipid-bound vesicles secreted by cells into the extracellular space, and some subtypes can include microvesicles or exosomes. They differ based on the biogenesis and release pathway, as well as their size, content, and function. Some of the common content of extracellular vesicles includes lipids, nucleic acids, and proteins, and they have been found to participate in cell-to-cell communication, cell maintenance, and in stimulating the immune response. So they can be sourced from a lot of different tissues or fluids, even plants, but the focus for this project is placental-derived components. So for this project, they're from amniotic fluid. And this is a schematic that is kind of the abstract of this project, and it shows how once the collagen type 1 is extracted from the bovine placenta and the EVs from the amniotic fluid, they can go through a channel of a microfluidic device where they get pinched into droplets that can cross-link at 37C after a couple hours. And this is basically the vehicle for releasing them into a wound environment for wound healing and senescence reversal. So a bioink combining both of these components would help with the structural support and to make the environment more responsive to repair. Moving on, this is the extraction for the collagen. The second picture shows the placenta that we received from our collaborators in the Department of Agriculture, UF, and we dissect it to keep the layers that we need for the extraction, homogenize it, and use an enzyme called pepsin, which digests most of the ECM proteins, but not collagen. So then after some rounds of centrifugation and NaCl precipitation, we get the extracted collagen that you see at the other end of the schematic. Then I went into the characterization of this collagen. So in panel A, you can see the DNA content, which can be a measure for cell contamination. And we wanted to compare the collagen that we extract with collagen widely available in the market, which is usually from rat tail. And as we hypothesized, from bovine placenta, our collagen is pure because the DNA content is around 2% of that of the market collagen, while the protein content is higher, about 1.5 times higher. And we wanted to confirm that this protein is actually collagen type 1. So in panel C, there's a Western blot with collagen type 1 antibody. And then in panel D, those are SEM images of the collagen fibrils after gelling to confirm the characteristic morphology, as well as type 1 antibody staining. Panel F is basically the gelled collagen, G is the lyophilized collagen, and H is when we reconstitute it to show that it can be stored and then reconstituted for later use after this specific extraction. Now, this slide is about the biocompatibility and regenerative capabilities of the collagen. And in the top half, there's a scratch assay with mice muscle cells that were allowed to attach on top of a collagen layer, which is commonly used for collagen scratch assays. And as you can see, the group labeled PC, placental collagen, at 0.01% concentration after 48 hours was fully healed, which is quantified in the graphs to the right. And then I also confirmed the biocompatibility through a Presto Blue assay in panel D, which also shows that the groups with the highest cell proliferation are the PC groups at 0.05 and 0.1% concentrations. And just to provide a rationale for why this regeneration is increased, we did ELISA assays for two common growth factors that contribute to regeneration, TGF-beta and VEGF. And both of these had a concentration 100-fold higher in the bovine collagen compared to the market collagen, while the endotoxin concentration, which is important for biocompatibility because they're remnants of bacteria, was decreased. Then moving on to the extracellular vesicles, first we wanted to establish the method of extraction that we wanted to move forward with. So we compared Exodus and UC. Exodus relies on microfluidic ultrafiltration and size-based sorting, and UC relies on density-based sedimentation under very fast centrifugal force. And basically, these are the two different EV populations that we got from the two different extraction methods. As you can see, the Exodus TEM image looks clearer, there's less debris, and that is also reflected in panel G, the purity graph, which is basically particles divided by protein concentration. And also in panels I and J, you can see those two growth factors that I mentioned before, their concentration normalized to protein content, which was the same for both. So when normalizing to concentration, it's the same, but since the purity was higher for Exodus as well as the particle concentration, we decided to move forward with this method for the following analysis. So for the following analysis, the EVs are from allantoic fluid, which is also in the bovine placenta. It's a sac connected to the umbilical cord that plays a role in waste storage, gas exchange, and vascular development in gestation. And this fluid is likely to contain even more growth factors and help mitigate cellular aging by supporting angiogenesis, cellular repair, and oxidative stress because of its location within the placenta. So we decided to use ETOs, which I previously said results in a pure population of EVs, for comparing the EVs from second trimester amniotic fluid versus allantoic fluid versus third trimester allantoic fluid. And these are the results, the TEM images again, which show that for the AF2 group, which is the earlier pregnancy-derived one, there's many more EVs, also reflected by the particle concentration graph. And for the bottom of this panel, there's another scratch assay comparing these AF2 EVs to being dosed to cells after a wound versus stem cell EVs and PBS as a control. And the group that has the lowest area after 48 hours is the AF2 EVs. So they're very regenerative. So then the next analysis we went into is for the anti-senescence capabilities of these EVs. This is a common staining done to test for the anti-senescent properties of different reagents. It's called SA-beta-gal, and it detects senescent cells by measuring the beta-galactoside activity at pH 6, which is a hallmark of senescence. So we treated trophoblast stem cells with the EVs that I mentioned before on day 4, on either just day 4 or both day 4 and day 5 versus the no treatment. And as you can see, the EV treatment day 5 group has the lowest expression of ASA-beta-gal. For the same cells, we also tried staining with gamma H2AX, which is also used to measure senescence because it detects DNA double-strand breaks by marking phosphorylated histone H2AX, which is a marker of DNA damage response. So the green color is the gamma H2AX, and the DRAG5 is for staining the nucleus for normalizing to cell number. And the group treated on day 4 and day 5, the purple group was the one with the lowest DNA damage per cell compared to the control. Now that I talked about the two different components and characterizing them, this is the part about the actual droplet encapsulation. So panel A has the microfluidic device that we use for this. It's based on a previous paper that my PhD mentor Samantha Lee worked on, and for collagen droplets. And in panel B is the labeling for each channel. One for the oil, one for the collagen and EVs that you want to encapsulate, and then the outlet channel for the generated droplets. Panel D shows what the droplets actually look like under the naked eye versus panel E under Gen 5 microscope. And then F is the cross-linked droplets after overnight cross-linking at 37C. The darker parts are the collagen fibrils, and then we also stained them to confirm that they were encapsulating the collagen with collagen type 1 antibody. So basically, the conclusions for this project is that we were able to do a high purity collagen extraction that yielded 1.5 to 2.5 milligram per ml of protein per 10 gram of tissue and is purer than commercially available collagen. It has a lot of growth factors, 100 times fold VEGF and TGF beta of growth factors associated with regeneration and lower endotoxin levels. So basically it's very biocompatible, which we also confirmed with scratch assays and Prestable assays, and the AFEVs also promote growth compared with stem cell EVs and attenuate gamma H2AX senescence marker expression as well as SA beta gal. We were able to encapsulate both of these components into a bioink that we generated using the microfluidic platform I talked about, and confirmed it post cross-linking with collagen type 1 antibody. And basically this resulting bioink is our hope for it moving forward is that it can help with structural support and targeted modulation of the wound microenvironment by shifting it towards a more youthful and responsive state, so it can speed up tissue repair, which can be helpful for very complex or chronic wounds. So these are my references, contact information, and acknowledgements to Dr. He and Dr. Minglu Yang's lab.
Great talk, Fabiana. And with that, we are right on time. So please direct any questions that you have for Fabiana to the chat. And thank you to all of our speakers for this session. You guys were wonderful. With that said, I'm going to direct this over to session 2's moderators, Christine and Francisco. So take it away from there.
I think Fran just got booted out. Here he comes.
Since Fran is not here yet, I would like to start the session.
No, Fran is here. He came back in. He's here.
Oh, he came back in. Okay.
Yeah.
Yeah, sorry about that. I was using, I don't know, like, thank you all for being here in this session too. That it's about the here. Going super slow. This biomarker discovery and therapeutic advances in women's cancer. Can you hear me well?
No, there's some
Can you
feedback.
hear me well?
There's feedback on your line.
By myself here. Sorry. Let me try to change this.
Okay, I should be okay, right?
Yes, you're good now.
Okay. Yeah, sorry about the delay. We're here in session 2. There is about biomarker discovery and therapeutic advances in women's cancer. I don't think I have to like remember like good practices, but please like keep your like microphones muted, and please do not record like anything. And yeah, it's my pleasure to introduce my co-moderator, Christine Wei from Michigan State University, and myself, Francisco Carrillo Salinas from the MIT. And yeah, if Christine, if you want to start introducing the first speaker.
Thank you, Fran, for the great introduction. It's my pleasure to introduce our first speaker of session 2, Hatun Dulan JT. She is a postdoc research fellow at Oregon Health and Science University. Today, she's going to talk about breast cancer liver metastasis, histopathological features at primary and metastatic diagnosis. If you have any questions in the end, please direct your question to the chat.
Can you see my screen? Hi,
Yes.
everybody. Thank you for giving the opportunity to present my work here today. Breast cancer is the most is the most common cancer type among females, and it's estimated that over 300,000 new cases will be diagnosed in 2025 in US. With recent advancements in early diagnosis and personalized targeted treatments, the 5-year survival rate for localized invasive breast cancer is over 99%. However, it reduces dramatically to 32% once the disease spread to distant organs. 11% of these young women would be younger than 45 years of age. Today, I'm going to use 45 as a cutoff for describing young women, and we use these in our lab. There is an increased rise in breast cancer in young women, and young woman breast cancer has poor prognosis. Young woman breast cancer diagnosis overlap with the childbearing years, and this is our focus point. Breast cancer diagnosed within the 5 years of last childbirth is defined as postpartum breast cancers. We have evidence that time since last childbirth impacts breast cancer survival. Today, I'm going to show you 2 human studies from our lab that supports that the postpartum breast cancer has high risk of metastasis. Here, in this is a multi-institutional study with 701 young women breast cancer, and cases delineated into 3 groups according to their parity status, and cases diagnosed with the context of recent childbirth has the shorter metastasis-free survival. And in this second nationwide registry, again, the cases diagnosed within the 5 years of last childbirth has shorter metastasis-free survival. All these cases were ER positive young women. With support of the ER status not being the main driver of distant metastasis in this younger individual cohort, the overall proportion of cases progressing to distant metastasis was the same regardless of the ER status. Postpartum breast cancer specifically has higher risk of liver metastasis, and in this cohort of our lab, in this study, we have compared 3 different cohorts of postpartum breast cancer with nulliparous never pregnant cohort on metastatic side, and we show that liver has increased metastasis when compared to lungs, brain, and bone in postpartum breast cancer. And this study also supports our results that the liver has 3 to 8 false postpartum liver metastasis in postpartum breast cancer cases. And why is breast cancer has increased liver metastasis in postpartum, not other the size of the distant metastasis? That's because there is a link between the mammary gland and liver during the reproductive cycle. During pregnancy and lactation, liver contributes to milk production of mammary gland with increasing its fatty acid synthesis and glucose synthesis. Upon the weaning, when the metabolic demands reduces, involution happens. Like mammary gland, liver increases its size and volume during the pregnancy and lactation, and when the weaning starts at involution day 2, weight starts to reduce. At the end of involution, the weight comes to nulliparous levels. And we have evidence in mice that increasing this weight is associated with increased proliferation markers, and decreasing in weight is associated with apoptotic markers. And we have also tested the increased risk of liver metastasis in our portal vein injection models of breast cancer. And we found that our results replicated our human findings that in postpartum, in early involution day 2, in 3 different cell lines, we had increased liver metastasis compared to never pregnant mice. Our lab was the first to describe winning-induced liver involution, and involution has a unique macroenvironment, and the stromal change during involution have similarities to describe tumor-educated metastatic niche, specifically on ECM remodeling and immune cell influx. And these attributes may contribute to high rates of liver metastasis in postpartum breast cancer. In our OHSU breast cancer liver metastasis cohort, we aim to identify the risk factors for BCLM by studying clinical and demographic data of primary cancers that metastasize to liver, and also to define the metastatic niche in BCLM with the goal of identifying treatment vulnerabilities. There's a clinical and histopathological attributes of BCLM has been poorly studied, and lack of BCLM cohorts with tissues is a barrier to progress. So we collected all eligible retrospective and prospective cases of BCLM between February 2019 and January 2024 with a total of 48 cases. We obtained BCLM biopsies and surgical resection tissues for FFP processing focus on tumor-adjacent normal liver interface to explore metastatic niche, and also collected the clinical and demographic data for BCLM as well as primary breast cancer diagnosis. In our cohort, we found that 38% of our cases were young women, and 57% of our cases were premenopausal, and 72% of our young women were postpartum breast cancers, which were all higher than expected US frequencies. And it has been reported that triple-negative breast cancer, HER2 positive breast cancer, high-grade and late-stage tumor has increased risk of liver metastasis. But in our cohort, we found that the majority of our cases were coming from luminal origin, and specifically almost 60% of them were coming from luminal A. And also more than half of our cases were were having low grade, and half of our cases were late stage tumors. When we made a subset analysis between the stage 1 and stage 3 cases, almost 70% of our cases were early stage primary tumors that didn't expect it to be metastasized to the liver. The hormone receptor status may change between the primary and the metastatic tumors of the same person is described as receptor conversion. And this is important in treatment decisions and lead to treatment failures in hormone-related therapies. In our cohort, between the primary ER positive cases, only 4 of our cases lost their ER status in their metastatic lesions, and also all ER negative primary tumors retain their ER status in the metastatic lesions. GATA3 is a marker of luminal breast epithelial cells and also tumors of luminal origin, and we found that GATA3 expression is strongly correlated with ER expression, and ER positive primary tumors retained their GATA3 expression, and in our ER negative liver metastatic lesions, the cases that have higher GATA3 expression, 3 of them were coming from ER positive primary tumors that this data is consistent with these metastatic lesions retaining their luminal phenotype in their mets. We evaluated growth patterns at tumor-adjacent normal liver interface to explore how the metastatic niche looks like in the interface and how the tumor progresses. International consensus guidelines described 5 different histopathologic growth patterns, and in our cohort, over 80% of our cases were replacement growth pattern. In replacement growth pattern, cancer cells grow along the existing liver the existing liver cell plates and effectively replacing the healthy hepatocytes and co-opting the pre-existing sinusoidal blood vessels. And this results in an infiltrative, poorly defined tumor border, which is associated with aggressive tumors. Here is a multiplex IHC staining of one of the replacement growth pattern cases in our cohort. This is HNE, and this is heparin staining the normal liver cells and GATA3 the tumor cells. We can see the poorly defined and infiltrative tumor border here. And this is also another case from our cohort with replacement growth pattern with, and also we know that collagen 4 and fibronectin are the two ECM components that line in the normal liver sinusoids. Here, we couldn't see a significant difference between the tumor area and the normal liver in the context of collagen 4 and fibronectin composition that is consistent that these tumors are using normal liver blood vessel sinusoids for progressing in the liver. As summary, our BCLM cohort is enriched in young postpartum cases with ER positive early stage disease, data consistent with newer large scale epidata. Our data suggests the classical prognostic criteria are less predictive for young women breast cancer liver metastasis. Within the liver, BCLM retained luminal characteristics. BCLM growth pattern was replacement, suggesting tumor cells use existing sinusoidal endothelium, and sinusoidal collagen 4 and fibronectin may provide adhesion and survival cues for invasive breast cancer. Thank you for listening to me, and thank you for all my lab members, our funding supports. Yes.
Thank you for a great presentation. If we have any questions, please direct in the chat area. If we don't have any questions at this moment, please still share in the chat area later. We will have Fran to introduce our next speaker. Thank you very much.
So our next speaker is Priyam Kumar from TAMU, and he's going to talk about discovering AI-guided targeted therapeutic for metastatic triple-negative breast cancer.
Hey guys, give me 1 second. I'm gonna share my screen here. There it is. Okay. Okay, can everybody see my screen? I'm just gonna minimize this, put this away. Give me 1 second. All right. Hi everybody. My name is Priyam Kumar. I'm currently a junior at the University of Pennsylvania, and this is a project I conducted in 2022 and 2023. The title of it is called Discovering a Novel AI-Based Targeted Therapeutic for the Treatment of Triple-Negative Breast Cancer Brain Metastases. All right, so first off, what is triple-negative breast cancer brain metastases? So TNBC is a subtype of breast cancer with the absence of 3 different receptors: estrogen, progesterone, and the human epidermal growth factor 2. Now, TNBC accounts for 10 to 20% of all breast cancer cases and affects 13 in every 100,000 US females. Now, the brain metastases of TNBC is a subtype that occurs in over 1/3 of all patients and is very, very deadly. So the goal of this project, here, let me move this. So the goal of this project was divided into 2 parts. First was we wanted to identify a target that is exclusively present or overexpressed in the TNBC BM cells. And the second part was to develop a therapeutic that could, 1, cross the blood-brain barrier, and 2, inhibit the overexpressed target. So the objectives of this project were divided into 4 different steps. The first part was to identify the target that was overexpressed in these patients and to explore the different parts of the target, such as the active site and what exactly are like the significant parts of the enzyme. 2 was we wanted to design a target-specific drug that used AI and other such as QSAR and other deep neural networks to kind of analyze the model and to find potential targets that could bind to this enzyme. Third part was we wanted to evaluate the efficacy of these potential therapeutics in these TNBC BM cell lines. And then 4, we wanted to validate these compounds in a mouse model. So the first step is we wanted to find a target that was overexpressed. So previously in our lab, we had done a lot of experiments with JAK3, and we found out that JAK3 was overexpressed in a lot of brain metastatic cancers. So shown here, just from like a qualitative assessment, we can see that with the cortex in a healthy individual compared to a TNBC brain metastatic patient, we see that JAK3 is clearly overexpressed. And so then that goes to the second part of our project. Here, excuse me. All right, so here we collaborated with a company called Atomwise Inc. And so we kind of gave them, we kind of had them like create a model of the JAK3 enzyme, which is shown right here in both the cloud model on the bottom and the kind of ball and stick model. And so here we wanted to analyze the active site and the molecular structure of this active site to find possible focal points for our inhibitor. And so here, it basically used a QSAR model. And yeah, and so here, then Atomwise, basically what they did is that they analyzed the active site of the enzyme and they sent us a list of possible therapeutics and compounds that could be used to inhibit this enzyme. And so in evaluating, so basically Atomwise sent us a list of about 80 compounds. And we wanted to see how effective these compounds were in inhibiting JAK3. And so first, we performed an in vitro kinase assay, which basically told us how effectively the compound inhibits the enzyme. So this is shown right here. I don't know if you can see my cursor. And so several of these compounds were shown to have a viable difference. And shown right here, compared to the control, C4, C17, and C19 were some of the ones that were shown to have lower the proliferation. And then, so sorry if it is loud in here also. So then we wanted to see to evaluate the potency of these compounds. So first, what we did is that we performed an IC50. Give me 1 second. It is a little loud in here. Give me 1 second. I am so sorry for this. Give me 1 second.
I think you're okay. At least I can't hear anything from your background.
Yep.
Okay, am I all good? I'm so sorry. There's like some people on the outside. They kind of just
You're
walked
okay.
in.
Yeah, no, we
Okay,
only hear
No
you.
problem.
thank you. Sorry, it was also affecting me for some reason too. But yes. Can y'all see my screen now? Is that okay? So then we performed the IC50 value, which evaluates the drug potency. Here, let me do this. And so what we found out was that basically lower the number, more potent the drug is. We found out that C4, C15, and C22 had some of the highest potencies of it. Then finally, we performed an MTT assay, which also showed us like the proliferation rate. And so higher the bar, higher the proliferation, lower the bar, lower the proliferation. And so from here, we can see that C4, C14, C15, and C17 were very efficient in reducing the proliferation in these TNBC brain metastases cells. We also then performed performed a permeability assay. So what we wanted, the reason we did this is because the most important part of this drug is its ability to cross the blood-brain barrier. Because how effective is a drug if it's not able to reach the target? And so I'm not able to zoom in all the way, but basically what we found was that these, even though C4 was shown to have great potency and was shown to have it was very viable in reducing the proliferation rate, it wasn't able to cross the blood-brain barrier. And so other compounds such as C14, and I'm not able to read that one right there, but they were able to effectively cross the blood-brain barrier. And so also all these experiments were done in the BRM813 cell line, which is a specific cell line of the TNBC-BM, of the triple-negative breast cancer brain metastases. And then in conclusion, this project was a success in designing and synthesizing a targeted drug for TNBC-BM. And we were able to test its efficacy in different cell culture models. However, the efficacy of these drugs need to be validated in more advanced mice models. And so that was the next part of our experiment. And so, but yeah, so I'd like to thank my mentor, Dr. Mishra, and the lab for her opportunity. And yeah, so that's pretty much my presentation. If you have any questions, I'd be more than happy to answer them. Also, apologies for me stopping in the middle. There was some stuff going outside that kind of distracted me. But yeah.
Yeah, no, don't worry about that. Yeah, if anyone has a question, can put it in the chat. I have a question in the meanwhile. So what type of data or structure did you use to predict like these models? Like did you use like established crystal structure of the inhibitors or
For which model of the JAK3 model itself, or the models of the inhibitors?
with the inhibitors?
So when we collaborated with Atomwise, what we did is that we sent them like our data and everything on JAK3, and using their models, they evaluated the active site and all the different domains and different like interactions, and they had a list of they had like a database of possible therapeutics and their structures, and they kind of sent us over a list of which of the possible ones would be able to fit into those active sites. I don't know if that answered your question, but it was a lot of AI tools are they're very complicated to sort of explain, and so that's why it makes it, I don't know, it made it a lot easier for us to use for drug discovery.
Perfect. Thank you very much.
I have one quick question potentially. Thank you for your
Yeah,
talk.
go
It
ahead.
was super interesting.
Mhm.
I was curious, and I really like to see that out of all of the targets that you tested, it was I wrote them down, the C14, 15, and 17. The C4 was able to have like an effect, but wasn't able to potentially cross the blood-brain barrier, which, as you
Yes,
said,
correct.
you really need that in order to have an effect.
Right.
So
Yes, ma'am.
could you anticipate any secondary or side effects from having those agents cross through the blood-brain barrier to the other brain cells, or like white matter? I don't know if you could anticipate any of that.
I'm not sure I'm able to anticipate any side effects, and so that's why the next part of our studies where we wanted to identify these in like mouse models to observe these things, because like one of the main aspects of, I believe, the C14 model is that it was a very small molecule, which made its viability in passing through the blood-brain barrier a lot easier. However, because of the size, it may have other adverse effects that we want to kind of like figure out more, like what exactly could happen. Like, although this is known to inhibit this enzyme, does it have any effect on any other biological processes that would affect the patients in general? I don't know if that answered your question. I hope it did, but yes, ma'am.
Perfect.
All right.
Thank you very much.
Thank you all for having me.
Okay, thank you for a great talk. I'd like to introduce our next speaker is Diva Priya Jaklabarti is a postdoc research fellow at Dartmouth Hitchcock Medical Center. The title of the talk is Evaluating Homogeneous Recombinant Deficiency, Microstellate Instability, and Tumor Mutational Burden Biomarkers in Ovarian and Endometrial Cancer Patients.
Can you see my screen? Okay. So thank you so much for the introduction and good afternoon, everyone. Today I'll be talking about these tumor biomarkers, homologous recombination deficiency or HRD, microsatellite instability or MSI, and tumor mutational burden or TMB, which are really important in the context of cancer, especially in ovarian and endometrial cancer. To begin with, I'm going to talk about some of the DNA damage and repair pathways that are associated with these biomarkers. So I'll focus on the homologous recombination repair and non-homologous end joining repair, which is mainly activated whenever there is a DNA double-stranded break caused by ionizing radiation and also reactive oxygen species and some chemicals. And the other pathway of importance with regard to this talk is the mismatch repair pathway that is activated mainly during errors of DNA replications, such as base-base mismatch or insertion-deletion loops. So most of these tumor biomarkers are detected by next-generation sequencing, whole exome and whole genome sequencing. And for homologous recombination deficiency, NGS is able to detect pathogenic variants in the homologous recombination repair pathway and also genomic instability, which I'm going to be talking about later. Tumor mutational burden can be measured by the number of non-inherited mutations in a genomic sequence, also detected by next-generation sequencing. And MSI or microsatellite instability looks at change in size of repetitive microsatellite fragments, which are sparsed across the genome and caused by defective mismatch repair pathway. So starting with homologous recombination deficiency caused by defect in the HRR pathway, as you can see in the figure here, both endogenous and exogenous sources can cause DNA double-stranded breaks, leading to activation of the HRR pathway through recruitment of proteins such as BRCA1, 2, PALB2, and RAD51. And these proteins are responsible for causing the homologous recombination repair. And individuals having genetic defects in any of these genes encoding the proteins are usually deficient for homologous recombination repair and are considered HRD positive. It is a predictive biomarker for multiple cancers, but primarily ovarian cancer, among other cancer types. And the HRD status of an individual, whether the individual is HRD positive or negative, determines their eligibility for treatment with PARP inhibitors or which is basically poly-ADP ribose polymerase inhibitors and also platinum-based chemotherapy. And on the right, you see the mechanism for this PARP inhibition. So whenever there is a DNA damage caused by a single-stranded break, the PARP protein is recruited and the cell undergoes base excision repair, which is another repair mechanism, leading to cell survival. However, in presence of these PARP inhibitors, just the olaparib, niraparib, etc., the PARP protein is inhibited and the single-stranded break moves on to become a double-stranded break. And in cells that are, or individuals that have proficient homologous recombination repair pathway, these proteins are activated, leading to double-stranded break repair. But individuals with deficient homologous recombination repair, or HRD individuals, basically there is genomic instability leading to persistence of the double-stranded break. And this is how synthetic lethality is induced in tumor cells through action of these PARP inhibitors. So now, as for detection of HRD, there are actually no single mechanism by which HRD is detected, and different labs use different techniques. However, next generation whole exome sequencing is the most commonly used technique for HRD detection, which looks at the HRR genes, the BRCA1, 2, and pathogenic variants in these genes, and also looks at genomic scars, which are large-scale genomic alterations leading to genomic instability. Now, some of these genomic scars are loss of heterozygosity or loss of entire chromosomal region within an allele, telomeric allelic imbalance balance or imbalance in copies of DNA at the ends of the chromosomes or telomeres, and large-scale transitions such as structural rearrangements that causes changes in copy number in large chromosomal segments. So HRD score is mainly determined as a sum of all these different genomic scars. As I mentioned, different labs use different approaches for measuring HRD. There are currently 2 FDA-approved tests for ovarian cancer for determination of HRD. One is the Myriad MyChoice test, which calculates the GIS score or genomic instability score as a sum of LOH-TAI and LST, and also looks at the BRCA1/2 status. The other one is the Foundation-1 test, which looks at tumor BRCA positivity, but just looks at the loss of heterozygosity as their measure for genomic scars. So there are currently many challenges with HRD testing, the first one being lack of standardization, since there are no single universally accepted definition for HRD. The current testing practices, both FDA-approved tests as well as laboratory-developed tests, do not test for variants beyond BRCA1 and BRCA2 for the HRR repair pathway genes, such as PLB2 and RAD51C are not looked at, and also epigenetic factors that can contribute to HRD, such as promoter methylation. And then another important factor to consider is the tumor sample quality and quantity, as inadequate tissue or low tumor content can affect HRD results. The HRD also has a dynamic nature where there are sometimes reversion mutations in the same HRR pathway genes that causes the patient to be resistant to treatment. So it has to be tested early before the treatment starts so that we get the exact HRD scores that we are looking for. And there are also clinical hurdles such as high cost and turnaround time that delays treatment decisions. The second biomarker of interest is tumor mutational burden, which is basically the total number of non-inherited single nucleotide variants per million bases in a genomic sequence. This is a predictive biomarker for multiple cancer types. However, it's according to the TCGA database, as you can see here, it's highest in melanomas, non-small cell lung cancers, etc. And you can see how variable the TMB scores are across the different cancer types. And for TMB detection, whole exome sequencing is again used. And then there is one FDA-approved test, the Foundation-1 companion diagnostics test for determining TMB status. In our lab, we use in-house NGS testing for identifying number of non-synonymous variants per million bases as a measure of TMB. And we exclude high variants which are highly frequent in common population databases such as NOMAD and also germline variants from our analysis. We include the interpretation criteria as scores less than equal to 10 mutations per megabase is considered TMB low, and greater than 10 is considered to be TMB high. And this is an evaluation study done in our lab where we compare data from Foundation Diagnostics with our own in-house NGS testing. So Augmet is our in-house NGS bioinformatics platform. And we looked at multiple different patients across different cancer types. And we saw that there was similar trend between the TMB high and TMB low individuals when we compared our data with the Foundation data. As for treatment and response, the TMB high individuals, usually the tumor cells produce neoantigens, which are presented to antigen-presenting cells and then are presented to the T cells or the cytotoxic T lymphocytes that have PD-1 receptor interacting with the PD-L1 ligand from the tumor cells, causing the T cells to be inactive and not being able to kill the tumor cells. However, checkpoint inhibitors such as PD-1, PD-L1 inhibitor, the FDA-approved drug pembrolizumab is able to act on the PD-1, PD-L1 interaction, causing activation of these T lymphocytes and acting on the tumor cells to kill them. The third biomarker of interest is MSI or microsatellite instability caused by genetic hypermutability or MLH1 promoter methylation, which affects the mismatch repair pathway. So individuals who have, whenever there is an error during DNA replication, individuals with proficient mismatch repair are able to repair the DNA by recruitment of these proteins. However, deficiency in the mismatch repair pathway causes acquisition of this mutation, leading to accumulation of replication errors and microsatellite instability. For MSI, the currently FDA-approved test is one based on PCR and capillary electrophoresis, the MSI diagnosis, MSI Oncomed diagnostics, and then there are next-generation sequencing techniques which are variable across labs. The FDA-approved test by Foundation One looks for MSI as well as TMB. And we have our in-house NGS testing where we look at MSI score as percentage of microsatellite sites showing instability. And we have a cutoff of 5%, where greater than equals to 5% is considered MSI high, greater than 1.6, but less than 5% MSI indeterminate, and then less than equal to 1.6% is MSI stable. There are also PCR-based MELT curve analysis, such as the IDILA MSI test in colorectal cancer for MSI high individuals. And currently, artificial intelligence can also predict MSI through digital pathology methods, but it has not been incorporated into clinical care yet. The clinical significance, MSI is mainly associated with colon cancer and MSI high, sometimes endometrial cancer. MSI high colon cancer may also be associated with Lynch syndrome, which causes increased predisposition to other forms of cancer. And there is currently FDA-approved immune checkpoint inhibitor therapy using pembrolizumab, which is used for treatment of MSI high tumors. And 80% of MSI high tumors are also TMB high. So it's the same drug basically used for both MSI high and TMB high tumors, but same is not the true for TMB high. So TMB high tumors are not always MSI high. So next I'm going to talk about an evaluation study that we performed in our lab where we compared 10 ovarian cancer and 1 endometrial cancer sample. The ovarian cancer samples were also tested at the Myriad My Choice, which is the FDA-approved test. And then the 1 endometrial cancer sample was tested at Foundation One. They have different cutoffs compared to what we set for our analysis that I'm going to talk about in the next slide. And we use 3 HRD controls: high positive, low positive, and negative. So we set a cutoff of 50 for our HRD positive cases. So individuals who are have an HRD score, which is sum of the genomic scars, LOHTAI and LSD, of greater than equals to 50 are considered HRD positive, and those less than 50 are HRD negative. And when we compared the data, we saw that most of these patients had concordant HRD results when we compared Myriad or Foundation results with our results, except for these 3, one of which actually had low tumor content, 20% tumor content, the reason for the discordance. The other 2 is maybe because of the variability in testing across different labs. And when we looked at the test performance, the positive percent agreement was 75% with an overall accuracy of 78.6%. And we also compared other biomarkers in these ovarian and endometrial cancer patients, and we saw that there was no correlation of HRD status with the MSI and TMB, since all these individuals had stable MSI and low TMB, even though they were deficient in HRD. So this brings me to the conclusions that due to highly complex nature of HRD biology involving multiple genomic events, there are variabilities in classification which could lead to discordant results. Although these tumor biomarkers mostly arise from pathogenic variants in DNA repair pathways, there are variabilities in the tumor microenvironment and across cancer types, underscoring the importance of comparing multiple biomarkers before taking therapeutic decisions in order to improve patient outcomes. In the future, we also want to expand our cohort for HRD validation. We have received an additional 30 samples, which we are going to add to the validation data set, and we want to progress towards offering this test clinically. With that, I would like to acknowledge my lab members and thank you.
Thank you very much for a great talk. Do we have any questions for our speaker? If not, I would like to ask a quick question. I would like to, I'm more interested in when you talk about the HRD, there may be a revision mutation. May I ask, in your knowledge, what are maybe the common cause? Is that from the therapy the patients receive?
Yes, it's sometimes from the therapy and sometimes just patient recovering. So it's they occur in the same genes that such as the BRCA1, BRCA2. So it's kind of adjacent to the original mutation, and it prevents the it causes treatment resistance because the cells become HRD proficient by acquiring the second mutation. So it's mainly due to therapy. So it's recommended that the HRD testing be done prior to treatment, so that we know that the patient is actually HRD positive.
Thank you very much. If we have more questions, you're more than welcome to post a question in the chat. And I'd like Fran to introduce our last speaker of the session.
Yeah, thank you, Christine. Our next speaker is Swati Suresh. She comes from University of Pittsburgh School of Medicine and is going to talk about investigating the role of PT1 CD200 axis and BRCA status in stromal modulation of TLS formation in high-grade serous ovarian cancer. Thank you.
Yeah. Can you see my screen?
Yes. Go ahead.
Yeah. Hi everyone. I'm Swati. I'm a PhD student. Thank you for the introduction, and also thanks for the organizers to give me the opportunity to present my work here. So I work with Dr. Kaufman, and I'll be presenting on this project that I'm working. To start off with, we know that high-grade ovarian cancer, or HGSOC, is one of the deadliest malignancies, and it's associated with 70 to 80% of ovarian cancer deaths. It's characterized by early metastatic spread, late diagnosis, and lack of curative therapeutics. Despite chemotherapy being a standard of care, the overall survival rate has not been improved in patients. And HGSOC is also strongly associated with BRCA mutations, where women with BRCA mutations are likely to develop HGSOC compared to non-BRCA patients. However, it's essential to understand the interaction between cancer cells and the tumor microenvironment that includes immune and the stromal cells. Our lab majorly works to understand the ovarian stromal microenvironment, and we found that normal stromal cells, or normal MSCs, are epigenetically reprogrammed by the ovarian cancer cells to have a cancer-supporting phenotype, which is called as cancer-associated stromal cells, or CAMSCs, with increased expression of WT1, which is a transcription factor. And our RNA-seq data also reveals that there is an altered transcriptome between normal and the cancer-associated stromal cells. And here is our flow data suggesting that cancer-associated stromal cells have significantly high WT1 expression when we compare it with normal stromal cells. And we also found that the CAMSCs provide an immune suppressive microenvironment, where, as we see here, CAMSCs regions have significantly high region where there is T cells being excluded, whereas the intratumoral TLS presence is very low in the stromal region. Hence, CAMSCs could be present as a potential target to combat ovarian cancer. So as I mentioned earlier, WT1 being a transcription factor and it's upregulated in CAMSCs, the main role of WT1, it's expressed during embryogenic organogenesis for proliferation and stemness, but the role of it is not much known in the ovarian TME. We used our RNA-seq data and found that WT1 was positively correlated with CD200 in the CAMSCs. So CD200 is an immune checkpoint protein, and it's a member of immunoglobulin superfamily that is upregulated in multiple cancers and including ovarian cancer. So the main function of the CD200 ligand is that it binds to its receptor on the immune cells and suppresses the immune activation. And there are various therapeutic strategies to target this axis. And currently there are clinical trials going on to enhance immunotherapy in cancer using CD200 immune checkpoint inhibitor. And as I mentioned earlier, women with BRCA mutations are susceptible to ovarian cancer. However, patients with BRCA mutations have better survival response. And here are some literature supporting this statement. However, when we add the cancer-associated stromal cell scores in the overall survival rate, we see that these high CAMSC scores negatively impact the survival benefits offered by BRCA mutations in patients.
So what drives this BRCA mutation patients to have better survival response? So we wanted to answer these questions, and we checked the BRCA mutant versus BRCA wild type patient tumor microenvironment, and we found that there is a presence of tertiary lymphoid structures in their TME in BRCA mutant patients. So TLS are clusters of immune cells that are present within the TME, and it's involved in producing humoral immunity against the tumor antigens. So basically, there are 3 stages of TLS, starting from lymphoid aggregate with clusters of just B and T cells, followed by follicular structures, and then later is the mature TLS that has the germinal center, which is involved in antigen presentation, somatic hypermutation, B cell proliferation, and class switching that is involved in production of antibodies specific to antigens. And one more thing is for the germinal center to be active, follicular dendritics play a major role. And we found that stromal cells have the capacity to differentiate into follicular dendritic cells under the influence of cytokines. And here is a figure from a HGSOC patient tumor microenvironment. When we did multi fluorescence imaging, we saw that this is the hallmark of a TLS. And then here we can see that it has the perfect germinal center in them. So when we also checked on the TLS presence, we saw BRCA mutant patients had increased presence of TLS size compared to BRCA wild type patients. And also the presence of lymphoid structures in BRCA mutant patients were significantly higher compared to BRCA wild type patients. And here is the representative immunofluorescence imaging, where BRCA mutant patients have increased presence of lymphoid structures compared to BRCA wild type patients. So overall, this explains that BRCA mutants have increased presence of TLS in their TME. Next, we wanted to answer the question, does CA-MSCs influence TLS formation and the overall survival benefits offered by the TLS? So we hypothesized that stromal cells would exhibit spatially distinct transcriptional states in relation to TLS proximity. And we found that TLS distal stroma had increased expression of markers specific to stemness using a digital spatial profiling analysis, whereas a TLS adjacent stroma had markers that has increased lymphotoxin beta, which is important for TLS formation. Next, in order to better assess the hypothesis that CA-MSCs are distal to lymphoid structures, we derived 13 gene CA-MSC signature based on a bulk RNA-seq data, and we found that these CA-MSC signatures were found to be highly expressed in the distant region when we compare it with the adjacent region to TLS. And also when we checked on the overall survival rate, we found that high TLS has better overall survival outcome in patients. However, when we added the high CA-MSC scores, we saw that this negated the benefits offered by high TLS. So next, as I mentioned, FDC, follicular dendritic cell differentiation is very important in mature TLS activation. And we know that we and others have shown that stromal cells have the capacity to differentiate into follicular dendritic cells. We wanted to see how the stromal cells differentiate and what is the expression of these FDC markers. So here is the morphology. So first we saw with BRCA mutant normal MSCs, BRCA wild type normal MSCs, and a BRCA wild type CA MSCs. And under day 10 stimulation media, we could see there's a change in the morphology under stimulation with cytokines, which resembles a follicular dendritic cell structure, whereas the CA MSCs did not have much difference in their structure morphology wise. Next, we did a multi-spectral flow analysis using different markers that are associated with FDCs. And we found that CA-MSCs have comparatively reduced expression of these markers when compared with BRCA mutant and BRCA wild type normal MSCs. However, comparing the normal MSCs, we saw BRCA mutant normal MSCs had increased expression of these markers. Next, we used one of our, next we wanted to answer, does knocking down CD200 in CA-MSCs impact FDC differentiation? So we used one of the CA-MSC line and knocked down CD200 using lentivirus. And here we can see that there is a 50% reduction in the CD200 expression after knocking down CD200. We used this line and performed multispectral flow with same FDC markers, and we saw that under stimulation media, these CD200 knockdown cells had increased FDC marker expression when compared with unstimulated media, suggesting that CD200 expression could influence the FDC differentiation, and knocking down CD200 can lead to increase in FDC differentiation marker. Further, we also wanted to specifically see the expression of CD21 since it's important important FDC receptor that is present in the FDCs that helps in presenting the tumor antigens to B cells for humoral immunity activation. So we use same CD200 knockdown cells and check for the CD21 expression. So we see that GFP positive CD200 knockdown cells here, and then we mask these cells to check for the CD21 expression. And then when we quantified the intensity of CD21, we saw that under stimulation, these CD200 knockdown cells had increased expression of CD21 intensity compared with unstimulated knockdown cells. So overall, these data suggest that CD200 influences the FDC differentiation, and it's important to target this for increasing TLS formation. So summary so far, we investigate the role of WT1 and CD200 interaction in enhancing CA-MSC's pro-tumorigenic function. We saw that BRCA mutant patients have increased TLS in their TME. And we saw that CA-MSCs negate the overall survival benefits that are offered by BRCA mutation and TLS presence in patients. And we also saw that CD200 knockdown reveals increased FDC differentiation and also CD21 expression upon knockdown. So next step is that we want to answer this question: what drives CD200 expression in CA-MSCs? So here we did pathway analysis using our RNA-seq, and we found that Wnt signaling pathway is significantly higher in based on our RNA-seq. And there is our enrichment plot showing that CA-MSCs have positive correlation with this pathway when compared with normal MSCs having negative correlation with this pathway. So next, here is our proposed mechanism and ongoing work. So we, based on this heat map, we were able to see that LEF1, which is a transcription factor involved in this Wnt signaling pathway, was highly expressed in the CA-MSCs group compared to normal MSCs. And we also performed a qPCR using the transcription factor LEF1 primers, and we saw that CA-MSCs had increased expression of LEF1 compared to normal MSCs. So here is our proposed mechanism, and we, from our preliminary research, we found that WT1 acts non-canonically to transcribe CD200 by the help of transcription factor LEF1. So our current goal is to study this mechanism. So overall, I would like to acknowledge and thank my lab and my collaborators. I would also like to thank my university for this opportunity. And then thank you everyone, and I'm open for questions.
Thank you very much, Swati. It was a great piece of work. Yeah, if anyone has any question, please put that in the chat. In the meanwhile, I have a question. So I had two questions, but you answered one with the last proposed mechanism. So I'm gonna say, so do you know if there is any, or have you used like an antibody against CD200 just to like in parallel with the knockdown of CD200 to see if the response is also blocked, like this immunosuppression?
Thanks for the question. So there is a clinical trial called with a drug called samalizumab, which we are planning to do it in a mice model and see how that is effective. But for now, we are working with the knockdown of CD200 using lentivirus, just with the in vitro cell lines.
Thank you very much. Looking forward for this clinical trial.
Thank you.
Are there any more questions? So in the meanwhile, I would like to say congrats to Christine. She just defended her PhD last week, so congratulations, Christine.
Thank you, Fran. Thanks, everyone. I would like to thank every speaker and attendee for a great session. It was very stimulating. We can take 5 minutes break and resume around 3:20 p.m. for our special session. And I think we just stay on the Zoom or yeah, okay. We'll stay and then come back around 3:20 p.m. Yeah.
Thank you very much, everyone.
Lisa, are we about ready to get started with the special session?
Yes, sir.
Okay. Well, hello, everyone. For today's special session, I have the privilege of introducing Dr. Samrita Dogra. She's a colleague and friend of mine from the University of Oklahoma Health Campus. Dr. Dogra completed her PhD in pharmaceutical sciences at OU in 2019 and shortly thereafter joined Dr. Bethany Hannafin's lab, where she has since studied the role of DCLK1 in chemoresistance and reoccurrence in ovarian cancer, which she'll be discussing today. So Samrita, I'll hand it over to you if you'd like to start sharing your screen.
Thank you, Cole. I hope my screen is visible.
Yes.
So thank you so much, everyone. And I would like to especially thank the organizers for giving me this opportunity to discuss my work, which is pertaining to the role of DCLK1 in mediating chemoresistance and metastasis in high-grade serous ovarian cancer. So I joined Dr. Hannafin's lab back in 2020, and that's where we started working on this particular kinase, that is DCLK1. And over the past 5 years, I feel like this project has grown tremendously, and we have generated really exciting data. And some of that unpublished data I'll be sharing with you all today. So, as you have already been listening to, that ovarian cancer is one of the most deadliest malignancy that is reported for women. And according to American Cancer Society, at least like in 2025, about 20,000 women are expected to receive new diagnosis, and of which 12,730 women would die of ovarian cancer. So ovarian cancer is considered actually as a silent killer because most of the patients, they are diagnosed with this disease when it has already spread to the distant sites in the peritoneum. And at this stage, the 5-year survival of these patients is really dismal. So epithelial ovarian cancer, it can either originate from the epithelium or from the non-epithelial parts. Among these, the epithelial is the most predominant, and it has multiple histological subtypes, of which the high-grade serous ovarian cancer is the most prevalent and also the most dangerous. So it is associated with mutations in BRCA1 and 2, as well as P53 mutations. So the biology of ovarian cancer is extremely unique. So what happens is that a majority of the patients, they experience peritoneal spread of metastasis, wherein the tumor cells, they slough off from the primary tumor, which is usually either in the ovary or the fallopian tubes. So these cells, they slough off either as single cells or as clusters, which are called as spheroids. And in the presence of the movement of the peritoneal fluid, as well as the accumulation of ascites over time, these spheroids, they go and attach to the peritoneal lining and the diaphragm and other distant organs in the peritoneal cavity, which results in establishment of metastasis. And one of the favored site for these cancer cells to metastasize to is the omentum, which is also the fat pad which is found in the peritoneum. We also observe some hematogenous spread or metastasis for this cancer, but it is relatively rare. Apart from this unique spread of metastasis, there is also accumulation of ascites or the fluid in the peritoneal cavity, which provides a unique tumor microenvironment. So this ascites that is collected from patients, it has multiple cellular as well as acellular components, which include cancer cells, spheroids, mesothelial cells, a lot of immune cells, and fibroblasts. And the acellular components, they include exosomes, like a lot of cytokines, adipokines, metabolites, and other growth factors. And this environment, it becomes really conducive to the extensive metastasis that is seen in these patients. So treating ovarian cancer is really challenging The first line of therapy for the patients is cytoreductive surgery, where they do surgical debulking, that is the removal of most of the tumor from the peritoneum, followed by use of platinum-based chemotherapy. Certain targeted therapies have already been approved recently, which include anti-angiogenic agents like bevacizumab, PARP inhibitors like olaparib, niraparib, and also recently antibody drug conjugates or ADCs. However, still there is a high proportion of patients that tend to relapse because of the occurrence of chemoresistance as well as extensive metastasis. Thus, there is a need to identify novel therapeutic interventions that can target these key players that orchestrate metastasis and the disease recurrence. So one of the players that our lab has been interested in studying is the DCLK1 or the double cotton-like kinase. So this is basically a serine threonine enzyme which exists in 4 different isoforms. So the isoform 1 and 2, it has this microtubule binding domain as well as the kinase domain. Whereas isoform 3 and 4, it lacks the microtubule binding domain, but still has this kinase function. So DCLK1 has been already established as a cancer stem cell marker in other cancers, specifically colon cancer and gastrointestinal cancer. Also, it has been shown to mediate drug resistance in pancreatic cancer. So in our experiments, we have seen that if we take samples from cancer patients, there is increased expression of DCLK1 compared to normal fallopian tubes or ovaries. And looking at publicly available data sets, we have seen that with high expression of DCLK1, the patients have poor progression-free survival as well as overall survival, indicating that DCLK1 is clinically relevant to be targeted in case of ovarian cancer. So there are various strategies that are available to target DCLK1, which include like a small molecule inhibitor, that is DCLK1N1. It is a competitive ATP kinase inhibitor, which targets the kinase domain. Also, our collaborators, Dr. Courtney Houchin, he has developed the DCLK1 targeted self-assembled micelle inhibitory RNAs, that is semi-RNA, and also monoclonal antibodies. And recently, Dr. Hanifin has been testing CAR-Ts also targeted against DCLK1 in ovarian cancer. So our published data in that we have already shown that DCLK1 is differentially expressed across ovarian cancer cells. So what we did is we grew different cancer cell lines under 3D conditions, and we looked at the DCLK1 expression. And as you can see here, that in most of the high-grade serous ovarian cancers, there was expression of the long as well as the short form of DCLK1. Interestingly, what we observed is that in the platinum-resistant setting, both the long as well as the short form was highly upregulated compared to the sensitive counterparts, indicating that DCLK1 may be important in mediating chemoresistance. We've also developed an in vitro invasion assay wherein we saw that I'm having some trouble with the videos. Yeah. So if we look at these tumor spheroids, we see that under controlled conditions, they are able to invade the matrix. However, when we treat these spheroids with the inhibitor of DCLK1, we see that there is decreased invasive phenotype, indicating that DCLK1 is indeed important for invasion. So quantifying this data, we found that even at 48 hours itself, DCLK1 inhibitor was effectively able to reduce spheroid invasion in vitro. So we tested this further in vivo, wherein we administered spheroids that were generated from platinum-resistant cell lines intraperitoneally in the mice. And we imaged these mice at regular intervals, and they were administered the DCLK1 inhibitor. Interestingly, what we found is that the DCLK1 inhibitor was effective in reducing the was effective in reducing the metastasis compared to vehicle-treated groups. So based on this background, we have hypothesized that these platinum-resistant tumor cells, they slough off from the ovaries either as single cells or spheroids, where they have high DCLK1, and this promotes the adhesion of these tumor spheroids to the mesothelial cell layer in the peritoneum, resulting in peritoneal metastasis. So to test this hypothesis, we used different model systems, including the CRISPR knockout of DCLK1 and platinum-resistant lines and overexpression of DCLK1 and OVCAR4 cells. To look at the impact of these cancer cells with their interaction with the mesothelial cells, we used MET5A, which is an immortalized lung mesothelial cell line, and also LP9 and LP3 cells, which are primary peritoneal mesothelial cells that were derived from ascites. Apart from this, our lab has also established ovarian cancer patient-derived ascites cultures from initial and recurrent samples that we also used for data analysis. Apart from this, we also used pharmacological inhibitor of the DCLK1 as well as mutations in the kinase domain that were overexpressed in the OVCAR4 cell lines. So looking at DCLK1 and its stemness property, we think that it might be associated with these metastasis initiating cells. So these metastasis initiating cells, they have certain properties, which include promoting EMT, proliferative potential, promoting invasion, adherence to the peritoneum, could be helpful in spheroid formation, and they also express certain markers like CD133, ALDH. So in our already published paper, we have already shown that DCLK1 tends to regulate EMT, proliferation, as well as invasion. So we wanted to see if DCLK1 checks the other boxes as well. So to test this, what we did is we looked at ALDH1A3 expression in these platinum-resistant spheroids, where we had knocked out DCLK1. And what we found is that with the knockout of DCLK1, the ALDH1A3 isoform, it was significantly reduced. These results were also consistent in case of the treatment of these spheroids with the DCLK1 inhibitor, as well as with the overexpression and the kinase dead mutants. Apart from this, we also looked at CD133 levels, and we found that with the knockout of DCLK1, CD133 was reduced. And here with the overexpression, we saw significant upregulation in CD133, which was reversed in case of the kinase dead mutants, indicating that the kinase activity is somehow important for maintaining these expression of these markers. We also looked at the patient-derived ascites organoids, from where we isolated protein, and that was subjected to protein sample, and we developed Western blots. So characterizing these ascites samples, we looked at EpCAM and Paxate, which are markers for epithelial cells, and most of these ascites samples were positive for these markers. Apart from this, we also looked at the DCLK1 long as well as the short variant, and interestingly, both in case of initial as well as the recurrent samples, we observed expression of both the long as well as the short variant of DCLK1. And also we found that ALDH1A3 was expressed both in the initial ascites organoid cultures as well as the organoid cultures that were isolated from the recurrent ascites patients. So we did a correlation between DCLK1 levels and ALDH1A3, and we found there was a positive correlation between DCLK1 and ALDH1A3, indicating that this is a clinically relevant observation. So based on this data, what we showed is that DCLK1 definitely is associated with the expression of these markers like CD133 and ALDH. Next, we looked into the other aspects or phenotypes that are associated with these metastasis-initiating cells. So we looked at the adhesion of the cancer cells to different extracellular matrix proteins in vitro. So here we used collagen, fibronectin, and laminin. And what we observed is that if we overexpress DCLK1 in the wild form, we see an increased adhesion to all the matrices, that is collagen, fibronectin, and laminin. And if we mutate the kinase domain, there was a significant reduction in attachment to fibronectin and laminin, indicating that DCLK1 is definitely important in attachment of these cancer cells to the extracellular matrix. So once these tumor spheroids or the cancer cells, they attach to the matrix, they tend to interact with the mesothelial cells. And these mesothelial cells, they tend to move away to allow these cancer cells to invade to the distal organs. So in fact, in case of clinical settings as well, we have observed that this is already like published data where they have shown that beneath the metastasis, there is no layer of the mesothelial cells. So to mimic this phenomenon in vitro, what I did is I grew the platinum-resistant spheroids, that is the DCLK1 knockout spheroids, and co-cultured them with the mesothelial cell layers and subjected these cells to live imaging through InQSight and observed the clearance of the mesothelial cells. So looking at these videos, you can clearly see that the tumor spheroids, they were effectively able to clear this mesothelial cell layer, which we see here in red. And the DCLK1 knockout spheroids, they were less efficient compared to the control spheroids. So upon quantifying this data, we see that the DCLK1 knockouts, they were significantly reducing the mesothelial clearance less compared to the control spheroids, indicating that DCLK1 is indeed important for interaction with the mesothelial cells in the tumor microenvironment. We also tested this with using peritoneal cells from the primary ascites. And here also, we observed that the cancer spheroids effectively cleared the peritoneal mesothelial cell layer compared to the knockout spheroids, which were less efficient. So this data gave us an idea that the cancer cells, they might be secreting something in the tumor microenvironment, which is affecting the migration of these mesothelial cells away from the tumor cells. So to test this in vitro, what we did is we collected conditioned media from the DCLK1 control as well as the knockout cells and co-cultured the mesothelial cells here on top of the transwell insert. And as you can see from these images itself, the control media promoted migration of the mesothelial cells. However, the knockout conditioned media was not effective in promoting migration. On the other hand, if we collected conditioned media from the cells that overexpress DCLK1, they were significantly stimulating the migration of the mesothelial cells, indicating that certain constituents that are secreted by these cancer cells, they are impacting the behavior of these mesothelial cells. Further, what we did is we took this conditioned media and co-cultured the mesothelial cells with this conditioned media and collected lysates. So since we observed increased migration, we wanted to look at the expression of these different epithelial and the mesenchymal markers. And what we observed is that in the presence of the knockout conditioned media, there was a significant reduction in the markers that are involved in more of a mesenchymal phenotype compared to the epithelial phenotype, indicating that the tumor cells themselves, they are promoting a EMT phenotype in these mesothelial cells. So going back to our figure, what we see is that in most of these phenotypes that we have, the data that I have shown, DCLK1 is promoting adherence to the peritoneum, and also it is modulating the interaction of the cancer cells with the components of the microenvironment, that is the mesothelial cells. So to delve further into what is actually happening between this intercellular communication, we looked at the tumor microenvironment. So basically these tumor cells, they secrete multiple factors, including cytokines, exosomes, and growth factors into the tumor microenvironment, which then impact the behavior of different components that are there in the microenvironment. It could be the immune cells, the mesothelial cells, or the fibroblasts. So our interest is more towards the communication between the tumor cells and the mesothelial cells. So to study this, what we did in our lab is we have developed this novel assay wherein CD63, which is a marker of exosomes, is fused into nanoluc. And this plasmid was transduced into these DCLK1 overexpression cells. So what we did is we used a luminescence-based assay to measure the CD63 positive NLUC activity, which indirectly indicates the increased excretion of exosomes into the media. So what we found is that with the overexpression of DCLK1, there was increased secretion of exosomes in the conditioned media, both at 24 hours as well as at 48 hours. However, the two kinase dead mutants, they were significantly able to reduce this exosome secretion at 24 hours, but however, this effect was not significant at 48 hours. So to further look at the composition of the conditioned media, we performed a cytokine array using conditioned media from the control spheroids as well as the DCLK1 knockout spheroids. And interestingly, what we found is that IL-6 was among the top most downregulated cytokine here that we observed in our cytokine array. So to further study this, we also performed a nanostring analysis wherein we had the platinum-resistant spheroids that were treated with the DCLK1 inhibitor for 24 hours. And we saw that transcriptionally, IL-6 levels were significantly downregulated in the OVCAR-8 CPR spheroids that were treated with the DCLK1 inhibitor. And this finding was also confirmed using qRT-PCR, where both the knockouts, they had less IL-6 mRNA expression compared to the control spheroids. This was also validated using ELISA, wherein I quantified IL-6 levels in the conditioned media, and we found that with the knockout of DCLK1, there was reduced IL-6 secretion. And on the other side, we found that the OVCAR4 cells that had overexpression of DCLK1, they produced a large amount of IL-6 in the conditioned media, indicating that tumor cells could be a major source of IL-6, which could be modulating the behavior of the mesothelial cells. So to further look at this particular pathway, what we think is that the tumor cells, they are the source of IL-6, which binds to the IL-6 receptor, which could be on the mesothelial cells, which in turn activates the JAK-STAT3 pathway, which could be promoting the mesothelial cell migration and also metastasis. So what we found is that in the control conditioned media, when the mesothelial cells are treated with it, we saw that the IL-6 receptor was upregulated, whereas in the knockout conditioned media, it was downregulated. Further looking downstream, we saw that the phospho-JAK2, it was upregulated in the cells that were treated with the control conditioned media, whereas in case of the DCLK1 knockout conditioned media, they reduced the activation of phospho-JAK2 in the mesothelial cells. And further going down, looking at STAT3 levels, we saw that there was a dramatic upregulation of phospho-STAT3, which was seen in case of the conditioned media derived from the control spheroids, whereas in case of conditioned media from the knockout spheroids, they were able to reduce it, almost take it back to the control levels. So this indicates that the IL-6 from the tumor cells is definitely activating or deactivating the IL-6 STAT3 pathway in the mesothelial cells, which contributes to mesothelial cell migration and the promotion of metastasis. So to summarize what I have shown today is that the ovarian tumor cells, they slough off from these ovaries, either as spheroids or as single cells. And we have these metastasis initiating cells, which could be a minute quantity of cells that have high DCLK1 overexpression, and they help in establishing metastasis. So DCLK1, it increases adhesion of the cells to the mesothelial layer, and the tumor cells, they release extracellular vesicles as well as cytokines like IL-6 in the tumor microenvironment and promotes mesothelial clearance, and which further leads to peritoneal metastasis. And if we genetically or pharmacologically inactivate DCLK1 using DCLK1 in 1, which is a small molecule inhibitor, we'd be able to block this whole process and reduce peritoneal metastasis. So the future directions that we are currently working on in
Samrita,
the lab is
I apologize to interrupt. I think you stopped sharing your screen on accident.
oh, it says I'm sharing my screen. Should I try again? Can you see it now?
Okay.
Okay. So what was the last slide that you saw?
I saw the summary slide.
Okay. So yeah, looking at the future directions, what we are looking at now is that to understand how DCLK1 regulates IL-6 and STAT3 signaling. Currently, we are working on in vivo studies using the DCLK1 knockout cell lines, looking at survival and its effect on metastasis. And further down the line, we'd be interested in profiling the exosomes to look at the contents that are secreted by the DCLK1 knockout or the overexpression cell lines. So in the end, I would like to acknowledge and my mentor, my current and previous lab members, our collaborators, Dr. Houchin, Dr. Bembrook, and also the funding from DoD and OCRA, without which this work would not have been successfully completed. Thank you so much for your patience. I'd be happy to take any questions.
Samrita, there is a question in the chat by Mindy, if you would like to read it.
Yes.
Do you want me just to ask it?
Yeah, sure.
Sure. Really great talk. I was curious in the intestinal epithelium, the stomach, and some other places in the body, DCLK1 is a marker of tough cells, and they have this chemosensory activity and stimulates immune cells. Do you think it's functioning the same way in ovarian cancer as well?
Yes, we do speculate that it might be involved in the immune response too, because there are studies that show that DCLK1 also is there in M2 macrophages, especially like in pancreatic cancer. They have done those studies. We plan to do like models using syngeneic cell lines like ID8, which are murine ovarian cancer cell lines. And then I think we'll be able to get like a clear picture of how DCLK1 modulates the immune microenvironment too.
Do you see any IL-25 in your model? Because tough cells specifically secrete IL-25.
So right now, the cytokine array that we had done, it had like a really limited number of cytokines that we were looking at.
Sure.
So IL-25 was not a part of it, but definitely once we are profiling exosomes or something like a unbiased screen, definitely we'd be looking out for that for sure.
Very cool. Thanks.
Yeah.
Hi, Dr. Dogra. I had a question. This might be more like a hypothetical, maybe. But since you were mentioning at the beginning in your background that the diagnosis of this ovarian cancer is so hard to pinpoint because the symptomology just comes so late after or during metastasis, my question would be, how would you predict to use DCLK1 to treat or target this tumor, given that the symptomology comes so late?
So yes, the thing is like what I feel or I believe in is that DCLK1 would be part of a maintenance setting that comes at like the later stage, not as a first line therapy or first line standard of care. So I believe like that would always be surgical debulking along with like the chemotherapy and the platinum stuff that goes on. But how we are seeing most of the effect in case of metastasis, I believe like yeah, it would be more of as a maintenance setting. Like, yeah, patients have been treated with that. And now we are using DCLK1 inhibitor in combination with those patients who are positive for DCLK1, either in their ascites or in their biopsies.
Thank you very much.
Yeah.
If we don't have any more questions at the moment, we can continue on to session 3, which will be moderated by Carla and Anu. If there are any more questions for Samrita, you can post them in the chat and she'll be present to answer them as they come through.
Thank you, Cole.
Thank you very much, Cole. I hope everyone can hear me. My name is Anu Balogun, and I'm from the University of Pittsburgh. And I'm very excited to co-host this session with Carla Martinez from Medical University of South Carolina. And the title of this session is Microbes, Metabolites, and Models of Diseases. And I'm personally very excited because this session is going to show us different and I guess diverse perspectives on how microbial activity and host responses shape health. And so with that, I'm very happy to introduce the first speaker, Subhomitra Ghoshal, and the title of her talk is Microbial Oxygen Consumption as a Driver of Anaerobic Niche Formation in Burn Wounds. So Subhomitra, if you want to share your screen and go ahead.
Hello, am I audible?
Yes, yes, you are.
Oh, okay, sorry. Okay. I hope the slides are changing.
Yeah, we can see your screen.
Can
Yeah,
you
the
see
slides
me?
are changing, yes.
Okay, cool. Okay, thank you so much for the introduction. Good afternoon, everyone. I am Subhomitra. I'm a second-year PhD student at the Mindy Engevik Lab from the Medical University of South Carolina. And today I'll be talking about exploring the dynamic microbial communities of burn wounds. Diving in, so the human skin is the largest organ of the body, which covers the entire surface, and it protects us against several microbial and cytotoxic threats. Our normal human skin is colonized by a diverse cutaneous microbiota, which serves specific functions like maintaining the skin barrier, controlling inflammation, and also in the wound healing process. But when the skin encounters some damage, it leads to a shift in the composition and the function of the microbiota. And burns are one such example of a damage, which is a severe insult to the skin. And these burn wounds are pretty vulnerable to several kinds of bacterial infections, which can be fatal for the patients. When a patient undergoes a severe burn, it often leads to the requirement of a skin graft. And these skin grafts are very much prone to different bacterial colonization, which can form biofilms, and which make the entire grafting process really difficult. In the clinic, we have certain procedures like using antiseptic irrigations and silver-based dressings, which are used to control these microbes. But both of these lead to a high amount of cytotoxicity and also inflammation. Surprisingly, the microbiome of burn wounds has not been well studied in the last 50 years because many of those could not be cultured using standard mediums. And the culture detection was also very low. So they no longer collected these samples in the clinics. But now with the advancement of technology, we have a very unbiased way to examine the microbiota, which is the next generation sequencing. It uses nucleotides and 16S and 18S ribosomal RNA and uses several methods and techniques to look at the microbiome. It is also a very time and cost effective way to have a look at the microbiome of these burn wounds. Which brings us to the aim of the project, which was to identify microbial composition of burn wounds and examine their bacterial interactions. So for this, we collected burn tissue samples from patients who needed grafting, And then we send those off for NGS. And we had 31 male patients and 9 female patients, a total of 40 patients. And for some, we did some repeated collections. So we had a total of 76 samples. And we had these tissues from various parts of the body which suffered extreme burn. And then we tried to look at the microbiome profile in these burn wounds. So we found a diverse microbiota. When we looked at the microbiome of these tissue samples, we found some pathogenic Staphylococcus epidermidis and Staph aureus, both of which are facultative anaerobes. We also found some Enterococcus faecalis and Pseudomonas aeruginosa, which are also facultative anaerobes. We found specific wound bugs like the Acinetobacter baumannii and Proteus vulgaris, which are facultative anaerobes again. But to us, we also found some commensals, that is the non-pathogens like Bacillus subtilis and Streptococcus mitis, both of which were again facultative anaerobes. But to our surprise, we saw a huge number of strict anaerobes. It was surprising because the skin, as we all know, will be constantly exposed to oxygen. But we found a large number of strict anaerobes like the Anaerococcus, the Bacteroidetes, Cutibacterium, Bifidobacterium, Finegoldia, Fusiobacterium, Parvimonas, Privotella. I would just like to give 3 examples of patient samples just to give an idea of how their microbiome of those burn tissue samples look like. The first was a 60-year-old male who had suffered a burn in the left lower extremity. So we collected the tissue, sent off for NGS, and we saw that his wound was dominated by Staph aureus. We also found very strict anaerobes like F. magna, Anaerococcus. The next was a female patient who suffered a burn in the thorax region, and her wound was dominated by Staph aureus, and Staph epi. Here again, we saw the presence of very strict anaerobes like Parvimonas, Privotella, Cutibacterium, and Fusobacterium nucleatum. The third example was again a female who suffered a burn in the thorax region, and her wound was dominated with P. aeruginosa. And we also found some strict anaerobes like Anaerococcus, Bacteroides privotella. So we find that along with the presence of facultative anaerobes, we find a large number of strict anaerobes surviving on these burn wounds. So we wanted to ask that whether these skin wound microbes could create an anaerobic microenvironment that allowed the growth of these strict anaerobes. And for doing that, we grew bacteria from plates into rich medium, then subculture into a chemically defined medium. Then we put them on a 96-well plate. And then we put one plate in a plate reader that could record the growth curve overnight, and the other plate into a receiver machine that measures the concentration of oxygen every 5 minutes for 24 hours. So this is a very good traditional bacterial growth curve. As we can see, the very traditional lag phase, the log phase, and the stationary phase. And so this is Pseudomonas aeruginosa's growth curve. And this is the oxygen quenching growth curve for Pseudomonas aeruginosa, which shows the concentration of oxygen with time. And as we can see that when Pseudomonas aeruginosa hits going to the log phase, it quenches oxygen and keeps it quenched over the 20 hours. So we see the same thing for Staph epidermidis. But for Staph aureus, we see that it does quench it when it hits the log phase, but once it goes into the stationary phase, the oxygen concentration goes up, so it loses the oxygen quenching capacity. We see the same thing for Enterococcus faecalis, that it first quenches oxygen, but after 6 hours, it loses the capacity to do so. Similar trend is seen for Enterococcus faecium as well. So I'm going to show you an example of certain oxygen heat maps, which is going to let us look at our upcoming slides a bit easily. So this is one bacteria where we see that high oxygen in initial time, but with time it quenches oxygen, so the oxygen concentration goes down and finally becomes an anaerobic environment. We can have a second type which does start quenching oxygen but loses the capacity, and so we see high oxygen after some point of time. And we can have a third category of bacteria which do not quench oxygen at all. So then we grew up a combination of bacteria to see their oxygen quenching capacity. And when we grew up proteobacteria members, we saw that they were pretty efficient in reducing oxygen, although Acinetobacter baumannii and Calciciticus, they lost their oxygen quenching power after 8 hours. But the rest could do it really well. And we also grew up some Klebsiella and Enterobacter, and we see the same thing. Also for skin bugs like Corynebacterium and Micrococcus, we also see that they reduce oxygen pretty efficiently. For Enterococcus species, we see that they reduce it for a very small amount of time and the oxygen concentration goes up again. For common cells like strep, we see they are not very efficient at reducing oxygen. And also for lactobacillus, they were not very good, excepting for lactobacillus acidophilus, which did quench oxygen after 8 hours. So we see that we find a range of proteobacteria members which are pretty efficient at reducing oxygen. So we wanted to see whether the certain facultative microbes could support the growth of strict anaerobes, and we actually did this like for the proof of concept. So we have Acinetobacter baumannii, which is a facultative anaerobe. We have Fusobacterium nucleatum, which is a strict anaerobe. We grew them first in single cultures, and then we co-cultured them in the presence of oxygen in order to see whether the presence of Acinetobacter would allow the strict anaerobe Fusobacteria to grow in the presence of oxygen. And you can really appreciate from this microscopic image that when we tag these bacteria, you can see that how these blue-green Fusobacterium nucleatum clusters around the facultative anaerobe Acinetobacter baumannii in the presence of oxygen. So from here we can say, or at least this is a proof of concept where we can see that how Acinetobacter baumannii can actually create a small microenvironment where it quenches oxygen and allows the strict anaerobe to grow. So from here, we conclude that on burn wounds or on the skin, we have certain facultative anaerobes which can quench oxygen for a small amount of time or for a longer amount of time and can help its strict anaerobe friends to grow and cause more damage to the already damaged tissue and thus cause delay in wound healing. And now we're trying to develop some transwell models where we grow human dermal fibroblasts. And then on top of that, we put our human skin cells and we let it grow for 21 hours and we create a air-liquid interface in order to make it differentiate and form the different striated layers of the skin. And then we plan to cause burn, the same kind of damage to these skin cells. And then we have decided on a burn wound bacterial community, which we're going to add to these burn tissue in order to mimic what's happening in these burn tissue samples. And then from these samples, we plan to look at the inflammatory cytokines and also look at the pathology, that is how there's been a change in the morphology or what exactly these community of bacteria are doing and for how long they can bring a change in the inflammatory cytokines in order to delay repair. I would like to thank my lab, the Mindy Engewick lab and all its members, and also my sister labs, that is Dr. Amy Engewick and Dr. Kristen Engewick's lab, and all my funding sources. Thank you for listening, and I'm happy to take any questions.
Thank you very much, Supamisha. That was a very, very interesting talk. We do have a question in the chat.
Yep. Sure.
If you'd like to read that and respond.
Okay. I cannot really see the chat.
Okay, I can read it for you.
Just later, yeah, that would be great.
Yeah, she says, great talk. I'm wondering if you had access to any other clinical data from your patient skin samples. If so, did you observe any notable correlations or differential abundance of microbes across clinical variables?
Yeah, actually we have data like, so different people suffer different level of burn. So as we know that people do get a cocktail of antibiotics then, so we don't really have data for every patient regarding what their normal skin microbiota was before and after the burn, but we do have some patient samples where we have collected data over time of their injury and like over time of their recovery phase. So till date, we have not seen any particular correlation saying that this patient had this much of burn, so we see this amount of this bacteria, no. But then after looking at 76 samples, we do see certain common bacteria that's how we decided on our bacterial community that we would treat the cells with, so that we have an idea that, yeah, these were the common ones from all these samples. And so we can look at it that way.
Thank you very much. Is there any other question for our speaker? I have a question.
Yeah, sure.
How do you think your findings will inform, treatments in hospitals, like for clinicians? Like, do you think like doctors should be, I'm guessing like maybe they should be more aggressive in their treatments that have these facultative anaerobes?
Yeah. Yeah. So, like you saw that certain ones were pathogenic, but we also had some common cells bacteria, which are actually good for us. So, like, it's a huge combination, right? So, like, people, like, obviously, there are like different kinds of people who would look at the tissue remodeling and how injury repair is happening and how immune cells are there, etc. But then nobody has really looked at the existing bacteria there because we always think that it's something which is coming from outside, right, for infections. But then this might actually give a new insight that there are things present right in that wound, which is allowing, the other strict anaerobes to grow and then cause a delay in wound healing. So maybe we can actually target these bacteria, which we think are not really harmful, but then they are doing something which is allowing other bad bacteria to colonize and grow and make things even worse. So yeah, I think that can be a nice way of looking at things.
Yeah, that was very interesting. Thank you very much for your
Thank you
talk.
so
If there are any
much.
more questions we have, please feel free to put them in the chat, and I'll hand over to my co-moderator, Carla.
Thank you, Anu. So our next speaker is Alisa Gutierrez from the Medical University of South Carolina, and her talk will be Metabolic Insight into Lactobacillus Responses to Breast Milk, Unraveling the Role of Hydrophenylactic Acid in the Gut.
Hi, can you hear me?
Yes.
Okay. And can you see my PowerPoint?
Yes, looks good.
Okay, awesome. Thank you for the introduction. I'll jump right into it. The neonatal gut is rapidly colonized by microbes soon after birth, and one of the major commensals is lactobacillus. Lactobacillus is commonly utilized as a probiotic for many good reasons in both adults and infants. And some of those reasons for being used as a probiotic include protecting against pathogenic colonization, as well as contributing to gut development and homeostasis. Lactobacillus also has a great effect in that it can colonize well throughout the GI tract. So, like many other GI bugs, Lactobacillus colonizes extremely well in the colon. However, Lactobacillus is very unique in that it colonizes the small intestine and even the stomach. And because of its widespread colonization throughout the GI tract, Lactobacillus is well positioned to support nutrient digestion, especially of food molecules that might not normally reach the colon. And with this nutrient digestion also comes the production of bioactive metabolites, which is the focus of today's talk. And with these lactobacillus metabolites, we had two major research questions that we wanted to focus on. The first one being whether metabolite production is different between different diets, especially between human breast milk and infant formula. And then the second major question was whether these metabolites could potentially affect intestinal health. We first utilized untargeted metabolomics to look at the metabolites of lactobacillus. So we have 6 lactobacillus species as listed here, which we selected because they're present in human infant stool. They've been utilized in clinical trials, and the last 2, L. reuteri and L. rhamnosus, are actually present in some commercially available formulas in the United States. And we took these 6 species and cultured them in a chemically defined medium called LDM4, which was supplemented with 10% volume of fresh human breast milk, infant formula, or water as a control. And then after overnight incubation in an anaerobic environment, we collected the supernatant and sent them to our collaborator, Dr. Thomas Horvath at Baylor College of Medicine, who handles our mass spec metabolomic side of things. And then using this metabolomics data, we were able to create these 6 PCA plots, one for each of the 6 species, where the clustering is colored by the dietary treatment. So the gray points are water, the blue points are showing the points for breast milk, and then the pink is for infant formula. And then for all 6 species, you can see that there is distinct clustering depending on the dietary treatment, where in some cases like L. acidophilus, we can see that the most distinct metabolomic signature was in response to formula, versus for other species like L. reuteri, we can see very distinct signatures for both breast milk and infant formula. And then I'd also like to highlight the numbers of metabolites per dietary treatment. So focusing on L. acidophilus as a primary example, we can see that the intersection of the Venn diagram shows that 10 metabolites were produced in all 3 media conditions, whether that be water, breast milk, or formula. And then unsurprisingly, 0 metabolites were exclusively produced in the water condition. And then there were 3 metabolites produced with infant formula. And then a much higher number of 22 metabolites were produced exclusively with breast milk. And this pattern of a high number of metabolites with breast milk was very consistent across the 6 species. And then in line with the idea that there's a lot of metabolites being produced in the context of breast milk, we also further found that a lot of these metabolites have been shown to be beneficial for the host. And one of these metabolites was primarily produced by L. johnsonii, and this was indole lactic acid. Indole lactic acid is anti-inflammatory in several different intestinal models, and we find that it's not produced in the context of water. It's produced at very low levels with infant formula, but most importantly, it's produced at very high levels with breast milk. And then another metabolite was produced only by L. reuteri, and this was called vicinic acid. Vicinic acid is a naturally occurring trans fatty acid that has been shown to suppress inflammation in rodent models of colitis. And likewise, we find that it was not produced in the context of water, and it was also not produced with infant formula, but it was produced at very high abundances with breast milk. And then finally, the last metabolite I'd like to highlight from this metabolomic study was produced by all 6 species, and this was called hydroxyphenolactic acid, or HPLA for short. And this is a tyrosine derivative that has been shown to be an antioxidant and an antifungal. And we find that, again, like I said, it's produced by all 6 species, but primarily in the context of breast milk, where only for a couple species it was produced at low levels with water and formula. And while it is known that it is an antioxidant and an antifungal, we were super curious as to whether HPLA could potentially be physiologically relevant in the intestines because this has not yet been reported. And so because HPLA is known to be an antioxidant, our first instinct was to test its antioxidant activities in an intestinal model. Now, one of the major functions of antioxidants is to neutralize reactive oxygen species, or ROS. And so we took an intestinal cell line, treated it with hydrogen peroxide as an example of ROS, and then we added HPLA to see if it could neutralize the hydrogen peroxide. We utilized H2D-CFDA as a fluorescent indicator of intracellular ROS, and as shown in the gray bars, we see that when we add 1 or 2 millimolar hydrogen peroxide, the intracellular ROS does increase. And then when we add HPLA, shown in the green bars, it significantly decreases the amount of ROS. And then we also tested the same experiment using raw 2647 cells, which are a mouse macrophage cell line. And we did this because immune cells are also very important in the gut. And we find a very similar pattern where when we add hydrogen peroxide, the intracellular ROS increases, and then this is brought back down when we add HPLA. And now, after looking at antioxidant activities, we also wanted to see whether HPLA could potentially mitigate inflammation. So we used the same model where we have the intestinal cell line treated with both HPLA and hydrogen peroxide to stimulate inflammation. And then we used qPCR to look at the mRNA levels of some of the pro-inflammatory cytokines, and we found that IL-8 and IL-1 alpha were significantly altered. So again, looking at those gray bars first, this is when we add hydrogen peroxide, and we do see some increases in both of these cytokines in response to 1 millimolar hydrogen peroxide. And then when we add HPLA, in the green bars, we do see a significant decrease in both IL-8 and IL-1 alpha in response to hydrogen peroxide. And then finally, we wanted to explore the effects of HPLA on gut barrier function. We utilized a functional assay that we call the Lucifer Yellow assay, where we seed a monolayer of cells on a transwall insert, and then on the apical side, we add our compound of interest, so HPLA, and Lucifer Yellow. Lucifer Yellow is a fluorescent dye that, depending on your monolayer permeability, can pass through, and then we can measure the fluorescence on the basal side. And we monitored the pass-through over the course of 24 hours, and if we focus on the gray points first, we do see that there is a baseline level of the Lucifer Yellow pass-through, indicating that our monolayer is not perfectly intact. However, when we add HPLA, shown in the green points, it does significantly decrease the amount of Lucifer Yellow that has passed through, thereby indicating improved barrier integrity. And following these functional findings, we then turned to qPCR to look at the mRNA levels of some of these tight junctional components. And we find that adding HPLA does increase the levels of claudin 1, claudin 3, and occludin mRNA levels. So in summary, we did find that breast milk compared to formula enhances levels of metabolites that may support infant development and intestinal health. And then we highlighted indole lactic acid as well as vicinic acid. However, there were a lot of other metabolites increased with breast milk, as well as potentially harmful metabolites that were decreased in the context of breast milk. And so if anyone in the audience is interested, we'd love to chat more about that. And then, of course, we highlighted HPLA as a potentially novel metabolite to study in the gut. And HPLA was produced by all 6 species, particularly in response to breast milk, and we find that it might be a key modulator of intestinal health by decreasing ROS, decreasing inflammation, and improving barrier integrity. As for clinical implications, the use of probiotics in infants is definitely on the rise. For example, there's a lot of infant formulas that contain probiotics, including lactobacillus. And then even out in the community, I'll meet a lot of people who have told me that they used probiotics when they had their babies. And so with this really popular use of probiotics, we should really ask, how can we optimize their use? And so the first assumption might be, well, based off of our data that breast milk enhances the metabolites that might be good for the host, then obviously we should just pair probiotics with human breast milk rather than formula. But this is very naive. It's not always feasible. Moms might not produce enough milk or donor milk is not available, and therefore formula remains a cornerstone in American families. And so we might shift our mindset to maybe identifying breast milk components that drive the production of these beneficial metabolites, and then maybe using these as prebiotics in formula, kind of like how we use HMOs or human milk oligosaccharides in formula already. But our lab is very interested in an alternative route where maybe we can identify some of these beneficial metabolites like HPLA, and then use these as postbiotics to add back into formula as well. And then with that, I'd like to thank everyone from our labs, as well as our collaborators, Dr. Katie Chetta and Dr. Thomas Horvath. And with that, I'll take any questions.
Thank you, Lisa. So are there any questions?
I actually have one.
Yeah.
Yeah, that was a very, very fantastic and interesting talk. Very, very insightful. And seeing as we just had a session, about women's cancers, there are a lot of mothers out there who can't breastfeed because they had to have mastectomies, after their cancer. So my question would be like, as you said in your concluding slides, do you think like we should maybe begin pushing for those kinds of metabolites to be looked into, made, and then included in infant formula so that maybe we can replicate some of these protective effects that we see in breast milk?
Yeah, there's a lot of reasons why we think that the postbiotic route might be more feasible than say prebiotics or probiotics. One of them is with probiotics, so you're adding live bacteria to formulas, and sometimes that can be it could be dangerous if the manufacturing policies are not quite very clean or very strict. And so there have been some cases where when you add probiotics to foods or goods like that, that there can be contamination. So sometimes parents are worried about having live bacteria in formula. And then with prebiotics, kind of like with what our data is showing, even if you add popular prebiotics like fiber or HMO to formula, it won't always drive the production of these beneficial metabolites. So in fact, the formula that we utilized had 5 different HMOs. And so clearly those HMOs are not driving the beneficial metabolites. And so we think it would probably be very hard to exactly identify what those components of breast milk are and then add those to formula versus identifying the metabolites after culturing is a lot easier. And so that's one of the reasons why the postbiotic route might be a little bit easier to study and implement.
Thank you.
Perfect. I do have another question, Alisa, if we don't have any other ones. So my question is, do you think that metabolites that seem to be beneficial in babies, like for example, this HPLA could be used in adults where they have some sort of gut disease like Crohn's or things like that?
Yeah, we're actually very excited to potentially test HPLA in like mouse models of colitis because we do think that this metabolite could be applied in inflammation, not just in infants, but adults as well.
Yeah, very interesting. Thank you.
Thank
Okay,
you.
so if anyone has more questions, please put them in the chat for Elisa, and I'll pass it on to Anu for our next speaker introduction.
Thank you very much, Carla. So our next speaker is Kathleen Rhodes, and she will be talking to us about the establishment of a novel model of pediatric type C HE in rodents. So Kathleen, if you're ready, please share your screen and take it away.
Awesome. Thank you so much. Let me get this up and going. Get my laser pointer ready. All right. Can everybody hear me okay?
Yes.
Awesome. Thank you. Well, thank you for the introduction. This is some really cool preliminary stuff out of my dissertation work. I'm a newly minted candidate as of August, so super exciting that I get to share this all with you today. So first, hepatic encephalopathy, or HE, I know not everybody here has heard of, so I'm going to get everybody oriented and then focus on the current understanding of pathology in the context of pediatric cases, then get into the aims of what we're trying to do with this model and why it's important that we establish it before getting into the preliminary data of establishing it, and then wrap up with some current conclusions, future directions, and the limitations of the proposed model. So hepatic encephalopathy, or HE, as I'm going to refer to it, is essentially brain dysregulation stemming from the liver when broken down into its two singular terms. This can begin by just the liver getting injured, and it results in a wide array of cognitive deficits stemming from shortened attention span, changes in learning and memory ability, mild confusion, and it can progress all the way into coma and even death. Up to 1 in 2,500 children in the US are diagnosed with a serious liver disease that can precipitate into hepatic encephalopathy. And of these, the most common cause in pediatric cases is biliary atresia. This is a deficient or not well functioning flow of bile based on either deformities of the hepatic tree or a lack of hepatic tree. So there's changes in bile flow, and that will lead to cirrhosis of the liver. In a small cohort of about 160 kids, this was done back in 2023, if I'm remembering right, 31.2% of those children experienced an episode of HE from a chronic liver disease. Widespread epidemiological studies are super, super limited. It's a very small field, which means that the long-term impact on a developing brain and what that might mean for long-term cognitive ability is very widely unknown. So as we currently know it, the way that it works is essentially you get an injury to the liver. Again, like I mentioned earlier, most commonly it's a chronic liver disease that leads to changes in liver function and liver activities. This results in one of the primary jobs of the liver, which is detoxifying ammonia from the blood. This is now impaired, so you end up getting hyperammonemia, or buildup of ammonia in the blood. This then impacts the brain and results in widespread neuroinflammation, which is known to decrease cognition, but has also been associated with potential cholesterol dysregulation, not necessarily in itself by just the hyperammonemia, but potentially bile acid signaling in the brain. Now, like I mentioned earlier, it's unknown if adult and pediatric pathologies are different, because this is how we understand it in the classic adult models of acute and chronic liver failure precipitated HE. We don't know in kids if it works the same, if there's potential protection due to higher levels of synaptic plasticity or what. There is the potential for irreversible deficits to cognitive ability to be sustained as a result. And current treatment strategies, there's no targeted therapeutics for hepatic encephalopathy at this moment for any type. And none of them fully address the cognitive deficits that are sustained. So the need for this model is incredibly high. We don't know how many kids are affected, but we know that there are some that are affected, and they deserve to know what's going on. So currently, there's only one slight animal model that we can use. It's a version of the bile duct ligation model for chronic liver disease. However, this is irreversible, which doesn't allow for long-term studies into cognitive ability after resolution of a liver damage, because we know that it's very rare that you're going to have a child with a chronic liver disease that no treatment attempt is ever made. So the goal of this is to have a reversible model where you can sustain liver damage, observe cognitive changes, resolve the liver damage, and then observe what happens long-term for cognitive ability. So how this works is we take corn oil and carbon tetrachloride. Carbon tetrachloride, it will make radicals and induce liver peroxidation, oxidative stress to induce chronic liver disease. It results in a lot of fibrosis, necrosis, inflammation, all characteristic of chronic liver disease. We give it to them via IP injection 3 times a week. This dose of 0.2 milliliters per kilogram is roughly 1/5 of the adult mouse dose of carbon tetrachloride. So it is adjusted for the pediatric cases. We start this at postnatal day 14. This is equivalent to a newborn to an 18-month-old baby, depending on what processes in the brain and what stages of development that you're looking at. Then we will continue treating them, assess their blood ammonia at week 3 to see if we're getting any changes or seeing those markers of liver damage like high blood ammonia. Then at week 6, this is equivalent to postnatal week 8. This is when both the male and female systems are developed, because we know that the male system is associated with some of those cognitive changes. We need that to be fully developed to be able to observe the impact. So then we begin behavioral assessments. We keep monitoring blood ammonia. And then at week 8, which is technically 10 weeks of age, this is equivalent to a 20-year-old young adult in humans. That's when we end treatment, and we will assess all of the liver damage. So first, we're looking at the level of damage to the liver with this model, making sure that the carbon tetrachloride is actually causing liver damage to give us cognitive changes. We looked at serum ammonia level, serum chemistries, my bad. And these are commonly seen elevated in human patients as well. They should be contained within the liver. Whenever you have damage, these will be elevated in the blood. So here we have serum AST and ALT levels. And in the corn oil groups, both show significant increases in serum chemistry levels. When we perform serious red staining, serious red looks at collagen deposits, so it's a way to identify fibrosis with this nice dark red coloring. So here in our vehicle groups, neither of them show any levels of liver fibrosis, but here in the carbon tetrachloride mice, there is positive serious red staining for fibrosis. Then we looked at the hyperammonemia and tracked it over time. So here we can see in the week 3, so they've been getting the corn oil and carbon tetrachloride for 3 weeks. Our carbon tetrachloride mice show significantly higher levels of blood ammonia. Again, after 6 weeks, they still have higher ammonia levels. And again, in the 8 weeks, our carbon tetrachloride mice have much higher ammonia levels. So showing that they have liver damage. Then we look at brain ammonia tissue. We utilize Nestler staining. Nestler's reagent uses mercury to precipitate out ammonia. So all of these black spots is where the ammonia has been precipitated out of the tissue for observation. So here in the cortex, the hippocampus, and cerebellum, we can see that in our vehicle groups, there is no ammonia present, as there should be. But in our carbon tetrachloride mice, we can see that there is hyperammonemia in key regions of the brain associated with cognition, learning and memory, and motor ability, which are all affected during hepatic encephalopathy. Then we're getting to the behavioral assessments here for the neuromuscular ability of the mice. So looking at that mind-motor-muscle connection, we can see that the grip strength of our carbon tetrachloride mice is significantly decreased compared to those vehicle mice. And then we also performed a balance beam assessment. So this is 2 days of training before the third day. That way it's not just a new test and they don't know what they're doing. They've been trained on how to do this. But we see that our carbon tetrachloride mice slip significantly more, so showing that there might be a deficient connection between the mind and the muscle. So here I'll show you those videos with the approval of the IACUC committee. I think I have to turn my laser pointer off. There we go. Okay, so here you can see, great, he's fast, super speedy, no slips, investigating, but not really deficient. But here in the carbon tetrachloride mice, watch those back feet. You can see that he's really slipping a lot. It's not a new test to him. He just has some issues between the mind and the muscle to be able to coordinate this activity really well. So showing that there is some deficiencies, but not that they're too sick to walk. It's just they're having issues. There we go. Let me get my laser pointer back. So then to wrap up neuromuscular ability, we also did a rotor-rod assessment. This is kind of like, if you guys have ever seen Wipeout, it's this little turning rod and you have them walk on it. And sicker mice typically will fall off a lot faster. So we record the time that they spend walking and the distance traveled. So here you can see that our carbon tetrachloride mice walked significantly less distance than our vehicle mice on the rotor rod, and they spent significantly less time, indicating overall that there is impaired neuromuscular ability, which is correlated to changes in the cerebellum. Then we looked at learning and memory ability. So here we utilize what's called a novel object recognition test. So this is another 3-day paradigm. You have 1 day where they're just getting used to being in the apparatus that we observe them in. On the second day, you expose them to 2 objects that are the exact same. That way they get used to it, they can sniff it, check it out. And then on the third day, you swap out one of those objects for a new object. And this really hammers on mice's innate natural curiosity to interact with things that are new to them. So here in our vehicle mice, we can see that really well illustrated. Here with the novel object, they explored significantly longer periods of time than the familiar object. Meanwhile, the carbon tetrachloride mice spend about equivalent amount of time, so showing that they don't remember that they saw this object yesterday, and they're spending the same amount of time with both. We turn that into a discrimination index to account for the fact that some mice might spend more time total exploring than other ones. So here you can see the vehicle mice with a more positive discrimination index indicates that they are exploring the new object much more than our carbon tetrachloride mice, which have a closer to 0 discrimination index, so indicating more equivalent time with the objects, which shows some level of impaired memory ability, but not necessarily an aversion to the object. When we're looking at neuropsychiatric status, we use the nestlet test. So here I have some guidelines showing how we score the nests. A mouse who builds a good nest is usually doing pretty well psychiatrically, and a mouse who isn't doing as well won't. So here you can see that our carbon tetrachloride mice scored significantly lower on their nest building, but they had no significant change in the amount of nestlet that they used, because one of the criteria is, do they fully shred the nest packet that they're given, or do they not touch it at all? And so it shows that they know how to start the process, they just can't finish it. Finally, for psychiatric status, we look at open field assessment. This looks at anxiety and depression-like disorders. So you have the inner field of the box, and mice that are not driven by more anxiety-like behaviors, they'll go into the middle of the apparatus. So here when looking at the time in the total apparatus, we can see that across the entire apparatus as a whole, the carbon tetrachloride mice actually travel a lot more, but there's no difference in how much time they spend in the inner zone compared to the vehicle, so less indicative of a neuropsychiatric problem with this model specifically. So then when looking at glial cells, we do know that neuroinflammation is impacted. Here we did IBA1 staining for microglia, and the number of microglia was significantly higher in the carbon tetrachloride group in the cortex, hippocampus, and cerebellum. Then we're looking at astrocytes marked with a glial protein. We can see that in all three of those same regions, there wasn't a difference in astrocyte count, indicating that maybe microglial recruitment might be a little bit more involved than astrocytes. So conclusions, we think that this dose is sufficient to induce liver damage. However, we've only ever done this in this strain of mice. We haven't tested it in rats. And there is the potential for cognitive deficits to be caused by the carbon-tet itself and not necessarily the byproduct of chronic liver disease. So we'll be doing more work to make sure that that's not happening. And we'd like to increase the cohort size to really get this validated before we go and publish it. And with that, I'd like to thank my awesome lab, my collaborators for teaching me how to get all into the behavior stuff. And I'll take any questions.
Thank you very much, Catherine. Is there any question for her?
Hi, Catherine. I do have a question. I think it's super cool to be developing this new model, especially when you explained in the background that there's only one model, but it's not a very good one. So I think it's pretty impressive. I did want to ask, and I might have missed this in your last slide with the future directions. You had mentioned in the beginning, you had mentioned at the beginning that one of the kind of hallmarks of HE is that you can like be reversed. So are you going to be testing either reversal or maybe not reversal of the damage, but kind of the getting better of the mice at some point? And like, what would that entail? Either doing those same readouts in those same mice after a longer time point? And would you be concerned that those mice would learn those behaviors and therefore perform better than what sick versus normal is?
These are all excellent points. So yes, one of the things that we want to look at is reversing the liver damage to see if long-term anything happens to the brain. So you're not still getting stuff being sent by the liver to cause more damage to the brain. I just wrapped up the first cohort looking at recovery behaviors. Thank you. It took a while. But what we've seen so far is that, yes, we did have to take into account that you don't want to do the same behaviors on that same cohort of mice that you previously treated. You want to treat them, not observe behavior, and then only observe behavior in the recovery period, because you will have that issue of, well, they've already done this before. Is this amount of time that we're taking as a recovery long enough for them to forget? Because yes, they will do better if they've already remembered how to do it. We've seen that in adult carbon tetrachloride mice on the rotarod specifically. So we knew going in that that was going to be something to take into account. But what I've seen so far is that with these mice, after they recover for 8 weeks, so they've had 8 full weeks. We know in adults, it takes 6 weeks to have the liver recover or show significant signs of recovery from carbon tetrachloride treatment via oral gavage, 6 weeks. So we did 8 weeks in these pediatric mice. So they were around 16 weeks of age total. And they still have some pretty significant changes to muscle memory coordination. So their balance ability is off. Their time on the rotarod, their grip strength ability, their learning and memory ability is also still pretty impaired, not significantly so, but it is. But their Nestlet tasting and their open field assessments looking at psychiatric status completely unaffected. So really cool stuff. I'm super excited about it.
So we do have a question in the chat. They say gut microbes are known to impact animal behavior. Is the microbiota changed in your mice?
That is a fabulous point. I would love to also look at that because with HE specifically, the other half of my dissertation work is looking at the brain bile acid signaling, and certain bile acids can be conjugated or changed in structure by certain gut microbiota. There's even some gut microbiota that can conjugate things like GABA onto bile acids, which can then enter the brain and participate in potential signaling that could induce some of those cognitive deficits. So I would also love to really look at the impact of the gut microbiome on cognition, both post-recovery and pre-recovery. I think that's a really good direction.
I have one question. So I just have to ask, as someone who studies the liver, do you think those cognitive impairments that you are seeing are from the fact that you've induced this hepatic encephalopathy, or could it just be general like hepatic or neurotoxicity from the CCL4? And how would you know if it's one of those things?
Yeah, so that's the hard part with any HE model, is making sure that whatever agent that you're using to induce the liver injury doesn't have its own circulating systemic effect. That's one of the things that we had to really watch out for, because we decided to do an IP injection of corn oil and carbon tetrachloride, but we believe that at this dose, circulating systemic carbon tet is not enough to cause such significant cognitive changes, rather than just having a severe impact on the liver, because the liver is the thing that's primarily metabolizing the carbon tetrachloride into the chloromethyl radical, that's inducing the lipid peroxidation, the protein alkylation, things like that.
Okay, that was very wonderful and interesting talk. If anyone else has any questions, we still have a couple more minutes. If not, then I will hand over to Carla to introduce the next speaker.
Thank you.
Thank you. So our next speaker is Wan Ye from University of Pittsburgh School of Medicine, and the talk is about glucocorticoid receptor deficiency impairs gestational beta cell compensation and contributes to gestational diabetes.
Good afternoon, everyone. Can you guys hear me clearly?
Yes.
Okay. Hi, my name is Xuan. I'm a graduate student at Dr. Herringer's lab at the University of Pittsburgh. Today, I'll present our recent findings on the role of glucocorticoid receptor in beta cell compensation during pregnancy and its contribution to gestational diabetes mellitus. So gestational diabetes mellitus, GDM, is defined as newly onset glucose intolerance in pregnant women without the previous diagnosis of diabetes. It affects up to 10% of pregnant women in the US annually. Although GDM usually subsides spontaneously after delivery, its presence leads to adverse health outcomes for both mothers and children. For mothers, it increases the risk of preeclampsia and the need for cesarean delivery, and it increases lifelong risk of type 2 diabetes by up to 7-fold. For children, it has been also linked to a higher risk of childhood obesity and metabolic syndrome in the adulthood. To date, the exact mechanisms underlying GDM are poorly understood, which hamper the effective disease management. Pregnancy constitutes a unique metabolic state in which the mother acquires insulin resistance, particularly in their third trimesters. The insulin resistance serves to reduce peripheral glucose uptake in the mother, sparing glucose supply for the fetus. Preclinical evidence showed that maternal islets would undergo more beta cell replication, and beta cell mass increased by 2 to 3-fold during pregnancy. Beta cells would also enhance their secretory function. These adaptive changes are termed beta cell compensation, which counterbalances the insulin resistance during pregnancy. Insufficient beta cell compensation leads to GDM. However, the molecular mechanism underlying beta cell compensation is not well understood. And this is what our lab has been trying to explore. So one of the candidates we are currently interested in is glucocorticoid receptor. Glucocorticoid GC is a steroid hormone produced by the adrenal cortex, and it signals through its receptor GR. Once being bound by the GC, the GR translocates into the nucleus and regulates the transcription of its target genes. In the clinic, it has been known that maternal plasma GC levels rise and peak in the third trimester. This timing coincides with the physiological induction of beta cell compensation, suggesting a potential link between the two physiological events. Furthermore, genetic variants of GR causing functional dysregulation have been linked to beta cell dysfunction and gestational hyperglycemia. This clinical data emphasizes the critical role of GR in beta cell function and glucose homeostasis in pregnancy. Therefore, the goal of our project is to elucidate the role of GR in gestational beta cell compensation and its contribution to GDM. So we first tried to examine the GR activity in beta cells during pregnancy. We found that the same as in the patients, the maternal plasma GC significantly increases in the wild-type pregnant mice. GI is a nuclear receptor, and it functions mainly via its trans activity. So by using immunofluorescence staining, we found there is more nuclear localization of GR in the pregnant versus virgin mouse islets, suggesting an increased GR activity. In these wild-type mice, we also measured islet size and calculated the estimated beta cell mass. Both were significantly increased in the pregnant versus virgin mice. So we confirmed there is an increased GR activity in pregnant islets alongside induction of beta cell compensation. To investigate the effect of GR on beta cell compensation, we generated a mouse model with beta cell tissue-specific GR deletion. We crossed the tamoxifen-inducible PDX-3 line to mice with loxP-flank and NATRIZ1 gene, which is the gene encoding GR. We injected tamoxifen for 5 straight days. 3 weeks after the last tamoxifen dose, we isolated islets and verified the knockout effect by qPCR. And overall, we achieved 70% of the knockout effect with this model. Because female homozygous beta cell GR knockout mice, they are infertile, we then used the heterozygous knockout mice for the following experiments. We performed IPGTT on these mice. We found that beta cell GR knockout mice maintained normal glucose in the virgin state, while they had impaired glucose tolerance during pregnancy. In the first pregnancy, they only had mild glucose intolerance. But based on the knowledge that multiparity exacerbates GDM, we then pushed them to the second pregnancy when they showed significant glucose intolerance. And meanwhile, we found significant reduction of glucose-stimulated insulin secretion in these pregnant beta cell GR knockout mice. So we concluded that beta cell GR knockout results in decreased insulin secretion and impaired glucose metabolism during pregnancy. Interestingly, throughout 3 consecutive pregnancies, we found that beta cell GR deficient dams had reduced litter size and higher newborn weight, as shown by the representative image here. This effect mimics macrosomia in the clinic, which refers to a condition where a baby is born significantly larger than average for their gestational age, which is a common complication in newborns of GDM mothers. To our knowledge, this is the first rodent model to replicate the etiology of clinical GDM. And this suggested beta cell GR depletion-mediated GDM not only affects maternal health, but also adversely impacts fetal development. To understand how GR deficiency affects beta cell compensation during pregnancy, we performed dual anti-insulin and anti-glucagon staining for the histomorphometry analysis. And we found a trend toward reduced islet size in beta cell GR knockout versus wild-type mice. And if we stratify the islets by their size, we found beta cell GR knockout mice had a significantly reduced number of large-sized islets with a diameter above 200 micrometer, as this islet shown in the representative image. The estimated beta cell mass was also significantly reduced in the beta cell GR knockout versus wild-type pregnant mice. And this finding was further substantiated by the Ki-67 staining, which showed beta cell GR knockout mice had minimal beta cell proliferation during pregnancy, as the percentage of Ki-67 positive cells out of the total islet cells was significantly lower in the pregnant beta cell GR knockout versus wild-type mice. So we infer that the beta cell GR deficiency impairs beta cell compensation for the pregnancy. Another finding we want to highlight is that beta cell GR deficient islets have a greater areas of alpha cells, as shown by the representative images here. We can see the average alpha cell portion within each islet, as well as the average alpha to beta cell ratio were significantly increased in the beta cell GR deficient pregnant mice. We also noticed the frequent dispersal of alpha cells in the center of islets, as shown by this zoom-in image here. Although the origin of these changes remain unknown, these findings suggest that beta cell GR deficiency not only affects beta cell compensation, but also causes disruption of normal islet architecture. We also investigated the secretory function of GR-deficient islets in the ex vivo setting. We isolate islets from pregnant beta cell GR knockout versus the wild-type mice and culture them in the media overnight. Then we challenge them with high-dose glucose. We measure the insulin content within the islets, as well as the insulin levels in the media. Since individual islets have different sizes, we normalize the insulin levels to the total islet proteins. And we found that beta cell GR deficient islets have a lower insulin content within the islets. And the baseline insulin secretion under low glucose, labeled as 0 minutes here, was not different between the groups, but the insulin secretion 30 minutes after high glucose challenge was significantly reduced in the GR deficient islets. The stimulation index, calculated as the ratio of insulin secreted in response to a high glucose challenge compared to the baseline secretion, was also reduced in beta cell GR deficient islets. So this suggests GR deficiency not only impairs beta cell mass expansion, but also impairs its secretory function. To further understand the mechanism behind impaired beta cell compensation due to GR deficiency, we isolated islets from beta cell GR knockout versus wild-type mice and subjected them to RNA sequencing for comparative transcriptome analysis. We found 231 upregulated genes in the knockout versus wild-type islets, whereas only 10 genes were downregulated, suggesting that GR mainly functions as a transrepressor in beta cells during pregnancy. And we looked through these DEGs. Then we found two main groups of genes that seem especially relevant to our phenotypes. First, we found many collagen-encoding genes, such as collagen 3, 5, and 6, as well as the ECM remodeling genes. To validate these findings, we performed serous red staining to see if there is more collagen deposition in the beta cell GR deficient islets. And we found that indeed there is increased collagen expression in the beta cell GR knockout versus wild-type islets, as shown in the representative image here. We can see the beta cell GR islets, knockout islets, had more red stained fiber strands surrounding these islets. Reviewing literature, we found very little is known about the roles of these genes in beta cells, but this finding correlated well with the disrupted islet architecture we already observed with the IV staining. Next major finding is that GR deficient islets had upregulation of genes that do not typically express in normal beta cells, and the false expression of these genes leads to beta cell dysfunction. Some of these genes have been previously reported as disallowed genes, whose expression is particularly low or even undetectable in islets than in other mammalian cell types, including genes involved in steroid metabolism, lipid transport, and glucose metabolism. Among them, we were interested in a gene called aldolase B in particular. We are interested in aldolase B based on that in a large human cohort study, the gain-of-function state variant in ALDO-B promoter region has been associated with reduced glucose-stimulated insulin secretion. Also, in a transgenic diabetic mouse model, ALDO-B was also reported to adversely impact insulin release. And our qPCR validated that ALDO-B mRNA is indeed upregulated in the beta cell GR knockout versus wild-type islets. So GR deficiency really drives the abnormal expression of this detrimental gene in beta cells during pregnancy. In conclusion, our project highlights the importance of GR in beta cell compensation during pregnancy. Beta cell GR deficiency reduces beta cell mass and function. Beta cell GR deficiency is associated with alpha cell dispersal in the core of islets and abnormal ECM deposition, along with the perturbation of normal islet structure. According to our RNA-seq results, we believe that GR primarily functions as a transrepressor in beta cells during pregnancy, serving as a housekeeper that suppresses genes with negative effects on beta cell function. And lastly, impaired beta cell GR signaling contributes to the development of GDM. So I would like to sincerely thank my advisor, Dr. Dong, my colleagues, collaborators, and my thesis committee members for their invaluable support during my PhD training. And I also want to acknowledge the support from the Cochrane Weber Foundation and Research Advisory Committee at UPMC Hospital. So with that, thank you all for your attention, and I'm happy to take any questions.
Thank you. Wonderful talk. Are there any questions?
Yes, I do have one. That was actually very interesting. I felt like very informative for women in general. But I did have a question. Did you observe any sort of like compensation or, some other kind of mechanisms from, for example, the alpha cells or any other kind of cells, I guess, like that in the islet, in response to your beta cell GR deficiency? Do you think any one of these other cells could be like compensating? Maybe there are other like paracrine effects during pregnancy. Is that something like, what do you think?
Yeah, that's an interesting question. So since we observed the increase in alpha cell area during the histomal function analysis, so we did quantify the estimated alpha cell mass in the same way we did for beta cells, and we found there was no statistically significant difference between the groups. And we do plan to measure the plasma glucagon levels since we really see that change. But according to our experience, glucagon levels do not always correlate with the alpha cell mass. So it's very likely that we will see no change as well. But there's something like that's something worth doing, and it has to be done. And we are really planning to do that. Yeah. And if we have available resources, we will also consider to do the approaches with higher resolution, like single cell or single nuclear sequencing, which will show the more likely to show the cell-cell interaction within the islets.
Wonderful, thank you.
Thank you. Any other questions? Okay, so if you guys have any questions, please put them in the chat and then I will pass it on for our next speaker.
Thank you.
Thank you very much, Carla. So the last but not the least for our microbes, metabolites, and models of disease session this evening is Catherine, who will be talking about Akkermansia enables persistent, I'm not going to even bother. I studied microbiology, but I forgot all these names. Colonization through the mucin-derived cross-feeding in antibiotic perturbed mice. Catherine, if you're ready, please take it away.
Thank you so much. Alrighty, so, sorry. So the human gut is home to millions of microorganisms, which we refer to as the gut microbiota. Within that gut, there are these organisms which are largely commensal bacteria, meaning that they are symbiotic and are not known to cause us harm. These bacterial communities perform a variety of important functions in the host, including secreting enzymes for digesting substrates inaccessible to the host, producing anti-inflammatory compounds that suppress epithelial pro-inflammatory responses, educating the immune system, generating these short-chain fatty acids which serve as fuel source for the gut epithelium, and repressing detrimental microorganisms. These all contribute to the overall health of the host. When we go on antibiotics, as used in a lot of Western medicines, we can shift this bacterial community, and that can create a window where pathogenesis can occur. So these pathogens can come and colonize the gut, and a pathogen is a bacteria that is not normally a part of the microbial community that causes a disease. The one that we focused in on was Clostridioides difficile, which is an anaerobic gram-positive bacteria in the phylum Bacilloida, and it is a motile bacteria. It's also a spore-forming bacteria, and it's particularly problematic because of the toxins which it produces. The toxins damage the gut by acting as ligands, which bind to the gut epithelium and alter its state. In the US, it causes 500,000 infections per year, and it's associated with 29,000 deaths. And if you are diagnosed within the age group of 65 or older, 1 in 11 diagnoses will lead to a death, and the treatment of C. diff results in $4.8 billion in excess of medical costs a year. The symptoms of C. diff infection are due to the presence of toxins within the gut epithelium and consist of persistent diarrhea, pseudomembranous colitis, which, as you can see here, are these pockets filled with the bacteria pooled in the colon, and in severe cases, it can cause toxic megacolon, which can occur and then lead to death. So when C. diff enters the intestine, it encounters a thick mucosal layer which protects the colonic epithelium. This mucus is made up of proteins with these long glycan chains attached to them. So at the top of the glycans, there are negatively charged sialic acid and fructose residues, which are very difficult glycan residues, only able to be removed by a couple of enzymes. And then along the mucus, there are also galactose residues, N-acetylgalactosamidase, and N-acetylglucosamine, residues in glycans, which could all potentially serve as food sources for C. diff. So in order to remove or free up those sugars, bacteria have to have enzymes called glycosyl hydroxylase, which are enzymes that catalyze the hydrolysis of those bonds in these complex sugars. And so we wanted to see, is C. diff able to free up those glycans, which they would feed on? And so we looked at all of the different known genomes, the 772 known C. diff genomes, to see if they have these enzymes which are able to free up these sugars. And we saw that they had none of the enzymes able to actually free up those glycan sugars from the mucus itself. To further validate our understanding of C. diff bacteria and its liking to feed on these glycans, we ran growth curves with C. diff exposed to different sugars and let it run for 24 hours. Here, the growth curves show that within the presence of these glycans, it supports the growth of C. diff. So this is with glucose over around a 24-hour period, and we saw growth with sialic acid and GalNAc, which are at the top of those residues. And then further down the chain, we can see that these glycans do support the growth of C. diff. So talking earlier, C. diff is a motile bacteria. So we wanted to see if it would chemotax or would swim to these known glycans if possible. So what we did was we grew up C. diff and we tagged it with a fluorescing agent, CDSEFE, so that within our chemotaxis assay, we could read if through fluorescent data, if it would swim to these glycan sugars. In the base of our chemotaxis assay, we had a media, and then in control in the top of these capillaries, we had that same media. And then in other capillaries, we had these glycan sugars such as GluNAc to see if it would have motility enhanced towards those. So then after an hour incubation period, we saw that C. diff will swim and chemotax towards these glycan sugars. So this data indicates that C. diff can sense mucin glycans and swim to them within the mucus. So in the mucus layer, C. diff can interact with other members of the gut microbiota, and we were really interested in which microbes were present with C. diff during the infection, because we know that C. diff isn't able to free up those sugars. So if there was potential cross-feeding that was occurring with another member of the microbiota. So we looked specifically at Akkermansia muciniphila, which we know is a mucus-loving bacteria. So it is freeing up those glycan sugars. To further verify our understanding of that known data, we ran the same genomic sequencing on all of the known genomes of Akkermansia muciniphila, which there's 127. And we can see here that all of those genomes have all of these sialidase, the galactosidase, these enzymes which are able to free up these glycans from the mucus backbone. The one that the genomes were lacking is N-acetylgalactosamidase, which is at the very base of the mucus chain or the glycan chain. So it's freeing up many of these glycans, and it's only really feeding on 2 of the known ones. So it's creating a very glycan-rich mucose layer. Because of literature that is pre-existing and out there, we are aware that Akkermansia muciniphila is upregulated in the presence of antibiotic treatment. So it would be in an environment where we would also see C. diff infection. So to address this question, we first grew up C. diff and Akkermansia muciniphila separately, and then grew them together to see if these bacteria can even co-culture and exist. here you can see in this media, which we have the falsely covered green rods is our C. diff, and our pink is the Akkermansia muciniphila. They are growing successfully together, so we know that they can cohabitate. And so what we wanted to look at is with this altering of the mucus, would it promote C. diff? So what we did was we grew up Akkermansia muciniphila in a media, and then we let it condition that media, freeing up all those glycans, and then we spun down and took off Akkermansia. And so we had this conditioned media, and so we used a control media that was not altered via Akkermansia muciniphila, and also grew up in this new conditioned media, C. diff. And then we ran qPCR to see if the genetic expressions of C. diff would change or be altered at all in the presence of these metabolites. So we ran 15 genes of interest, but there were 3 very notable genes which we wanted to point out. We have CWP84 and FVP68 were both upregulated, which are both components that show that the durability of the build of C. diff was upregulated via these metabolites. And then we also saw this flagellal genetic component was upregulated in those that were grown up in the media, which was conditioned with the Akkermansia metabolites, which was leading to the construction of the flagella, which shows that it promotes genes promoting the durability of the bacteria and also the motility of the bacteria, which we know are 2 key components to this infection. We also wanted to see if it would alter the chemotaxis assay, if it would increase the chemotaxing of the bacteria. So we did one with the control media and then one with the conditioned media, and we saw an upregulation in the fluorescence of that was exposed to the conditioned media towards the GluNAc, which we saw earlier is a glycan that it does like to swim towards. So as mentioned earlier, an aspect of C. diff which leads to the infection is these toxins, which are altering the gut epithelial layer. And how this works is they do it via binding to the receptors as ligands, and these toxins are endocytosed and avoid the endosome, and it inhibits the actin cytoskeleton and causes this very distinct cell rounding.